Good morning to all participants. With great pleasure, on behalf of Bruker and Industron Nanotechnology Private Limited, I welcome you all for today's seminar on nanomechanics for biomaterials and soft matter. All participants are muted by default and will encourage them to keep it in mute during the talk. Participants will have the opportunity to interact with the speakers directly at the end of the talk. Please leave your questions during the talk in the Q&A tab window or in the chat box, and we will take them at the end of the talk. It is indeed great pleasure for us to be the part of this event and would like to thank all our participants for sparing the time for this event. Let me quickly take you through for today's program. We have 4 talks from starting with a keynote lecture from Professor Kantish Balani from IIT Kanpur. Then we have an invited talk from Professor Naveen Kumar from IIT Ropar. Then we have a talk from Dr. Syed Asif from Industron Nanotechnology Private Limited and Dr. Yogesh Jairam from Bruker India Scientific Private Limited. It's a very packed program, and we'll be focusing on nanomechanics for biomaterials and soft matter. So without further delay, I would like to introduce our first keynote speaker of the day. Professor Kantish Balani is a professor and head of Advanced Center for Material Science in the Department of Material Science and Engineering at the Indian Institute of Technology, Kanpur. He has graduated with his PhD degree in Mechanical and Materials Engineering from Florida International University. His major area of research are into biomaterials and coating scaffolds, energy materials, nanomechanics and nanotribology for biocomposites, and aerospace materials for ultra-thin and ultra-high temperature materials. He's associated with IIT Kanpur as a full professor in Department of Materials Science and Engineering. He's also a recipient of several fellowship and prestigious awards such as Class of 2021 Fellow by ASM International, 2018 ASM IM Visiting Lecturer awarded by ASM International, Swern Jayanti Fellowships 2016-17 by Department of Science and Technology by Government of India. So without further ado, I would like to invite now our Professor Kantish Balani. So Professor Kantish Balani, the stage is yours now.
Thank you, Dr. Pratyan. I'll start sharing my slide and please let me know. Is it visible? Shall I go ahead?
Yeah, we can see the slide.
Okay, great. So very good morning, everyone. And I really thank Dr. Pratyan for inviting me for this to give this particular keynote lecture for the workshop Nanomechanics for Biomaterials and Soft Matter. So today I'll talk something along the lines of decoding adhesion mechanics of bacterial proteins on biosurface. This may look very, biologically oriented, but it also has a lot to do with biomechanics. And that part I think you will understand as we go along. So I'm a material scientist, but then there is always a interface with other engineering disciplines. So I'll just let you through this. So I'll start with biomaterials and associated with some biobacterial infections, and then how do we go about developing antibacterial biomaterials. And we also have seen some interaction of S. aureus, which is a gram positive bacteria with certain biosurfaces via atomic force microscopy. But in between, I also will highlight the importance of nanoindentation. And then we also have seen the effect of silver and zinc oxide to translate them into the real life acetabular liner. And finally, I'll draw the conclusions. To start with, biomaterial is very generically defined. It's a non-viable material used in a medical device intended to interact with biological system. So this is a very stated definition, but there are some hidden parts which I think I'll probably highlight here. Let me use a pen, so marker, so that I can. Okay, that's okay. So yes, a non-viable material. I'm sorry, I'm not able to use the marker. So it says it's a non-viable material. But apparently, with time, things have changed. This is a definition by Williams in 1987. But now, once we make it also viable material, like maybe using some organ transplant or maybe some 3D scaffold, which already has cells on it, so it also has some viable entity into it. So we can make it more generic by saying it as also including viable materials. And again, they're used in a medical device intended to interact with biological system. The only thing is we want to make it more encompassing. So this is a very old definition, but still widely accepted. Then we talk about biocompatibility, and biocompatibility also looks very generic, but it's the ability of a material to perform with an appropriate host response. And again, this is very important in a specific application. Why I'm underlining these two terms is because an appropriate response means that it should not, once you implant something into the body, it should not start leading to blood clot or infection, or it should also have a normal healing time. At the same time, it's for a specific application, because the duration, if I take a syringe and maybe I can take my blood out and then everything is fine, but if I keep that material in my body for long enough duration, it may cause some problem in the body. So again, for dialysis or like for hip implant may stay in the body for lifetime, right? So I'm also mentioning that the duration also is very important. Something which is not toxic in a shorter duration may become toxic in a later term. Also, it might, something may clog in our liver, kidney, brain. So that also we have to take everything into account. So we keep talking about biomaterial biocompatibility. One thing is for sure, that safety is mandatory. Yes, we would like to restore some functionality, but it should not be at the cost of causing a harm to something else, right? So safety is mandatory. It should not become toxic that the person itself, I think, is not able to live further. How things have started? Things have started much earlier, 1940s, 1950s, when it was during World War time. So main thing was, to retain bioinertness. If someone has lost their limb during the war, they will just replace it with something which is workable. Just maybe a limb with a wood or anything that is only workable. So that aspect is now taken accounted as a bioinertness. Second aspect is bioactivity, which came about later in. So 1960s, 1980s, then the goal was to also induce some activity. So by triggering something, so it may lead to bonding with the bone, maybe able to release the drug. So that started sometime around 1960s, 1980s. So from bioinertness, we went to bioactive. And then later on, it went on to tissue regeneration. Like you might have seen this movies of Spider-Man, Dr. Lizard. The person is able to, give me a second, I'm getting a call from my home. Please give me a second. I'm sorry for this. Give me a second, please. I'm sorry, I thought there was something urgent. I'm really sorry for that. Sorry. So something like Dr. Lizard. So it was about generating that organ itself, and that's how this third generation, things have come to regenerate the entire functional tissue, and that's how I think things are going forward. So that now we are going from bioinertness to bioactive to now regeneration itself. But doesn't mean that we don't require bioinertness, we don't require bioactivity. It means that we require everything, but something can add. We can add functionality as we go along. And again, we can classify the biomaterials based on either they are which type of material, which can be polymer, metal, or ceramic, or it can be based on material response, like I just mentioned about bioinertness, bioactivity, bioresorption, or even toxicity. That is second class. And third part can be based on application, which can be, either structural or non-structural, which means which is bearing a load and non-bearing a load. So like for something for the contact lens, it is non-structural, but like for hip joint or something which is taking load becomes a structural component. But overall, it is a multidisciplinary field, because anything you want to bring it to market has to undergo the required in vitro tests, in vivo tests, then maybe some preclinical, clinical trials, and then only it has to be commercialized. Also, there might be legal aspects, ethical aspects, development aspects. At the same time, all people have to come together, work together. If there is a sensor, an electrical engineer may work with a biomaterial or a bioscientist, or even a material scientist. So that those things actually make it more interdisciplinary. Also, doctors have to come in, lawmakers, ethical policy makers, so those all have to come together and work for it. And just to take you to the market, so there are, I think, 2.9 million globally these are available, and around 70 to 80,000 surgeries are annually done in India, only in India. And the cost of surgery in India is much lower. That's why I think they're also called as a hub for undergoing this medical treatment. It is a lower cost, but the reliability is not that great. So still, I think in Gujarat and all, they still get this extruded rods, from abroad, because the reliability they actually sustain much longer. And if you look at all those components by Zimmer, Stryker, Corin, Elite, and all that, so or Surgival, so apparently they will come up with something to the surgeon and say, oh, this has added functionality. No manufacturer will say, oh, my product is inferior to someone else. They will always say, oh, I have something added, something more to this. So for a surgeon, sometimes it is not a very informed choice, because they will go it because they also probably get a cut or maybe some subsidy in terms of getting that implant to the patient. So I'm just telling that things are not straightforward as such. We would like to increase the functionality, improve the function at certain location, ensure that this implant binds the surrounding bone and also does not fail. So at least it should survive for 10 to 15 years at least. So for 7 years plus, the survivability is typically 90%. It means that at least 90% implants survive even after till 7 years. We want to make it much longer, so that people with those who are implanted much earlier, they also they don't have to undergo surgery again and again. So just to let you through, so there are different components of acetabulum. Then you have a liner, which is a polymeric, and then femoral head, which can be metal or ceramic. And this is the typical joint which you typically get to see, and that's how it is placed. I will just highlight one of few things. So complexity is arising because this liner what we're using is polymer. Why can't you use it metal and ceramic? Maybe I can tell you in a few minutes. Also, you need to ensure the sphericity. You need to ensure that there is good bioactivity. The stem should have a good bioactivity because it binds with the bone. And acetabulum component also should have a good surface coating so that it survives much longer. I will highlight one thing. So this component, I am saying that femoral head and a liner. This is the portion which will undergo articulation, this portion. And then it will join with acetabulum component, and on the top you can also, because it is again meeting with bone, so you can always have to do a bioactive coating. And this portion also of the stem, which also binds the bone, also needs to have some bioactive coating. But if we see this particular part, femoral head and the liner. So here we have a metal or a ceramic against a polymer, which is very strange. I can always use a metal and metal, that might be better. Ceramic and ceramic, that might be even better, right? So in real life, if I want to go for ceramic and ceramic, that will give me the best or highest life. But society will not accept it. The reason is, but with every step you will take, if you have this implant, it will give a screeching sound, chee, 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 chee. So I think that is not acceptable, right, in society. So next best couple will be metal and metal, but metal debris is again toxic. So again, that becomes a problem. So typically either if you have a polymer, then only polymer will actually come out as a debris which our body can somehow handle. So I would like to have a metal and a ceramic, a metal and a polymer, or a ceramic and a polymer. And now imagine the hardness difference is so high. So I would not again like to use metal, because again, if there is by some chance metal debris comes out, that again is not good, or metal starts to leach out. So that region also will have some similarity, some body fluid. So I'll keep it as a ceramic and then a polymer. So if I have a very hard and a very soft material, then it also is a issue. So I would also like to add some reinforcement and increase the hardness of that liner. Anyhow, so if I have this step, typically if I just implant it, there is a bone cement called polymethyl methacrylate, and then we glue it. But after a few years, it starts to loosen out, and then basically it again requires a revision surgery. So idea is to add a coating or bioactive coating around it, like hydroxyapatite, and when you insert it, then it forms a binding with the bone, and then you don't require that surgery because now it is bound to the bone. So this coating of hydroxyapatite also helps a lot in developing a better implant. So I basically work on this area, that I working on the acetabulum part, working on the femoral head part, and also depositing some coatings on the femoral stem. So I work on all the three areas. So this is so I'm anyway. So I'll try to take some focus, because now we are using metal We're also using ceramic as a coating. We're also using metal as an implant, polymer. So we use all kinds of material, right? So I'm just telling the requirement of materials is very, very high, though I work on biomaterial. And there are various aspects to it. I would like to make a shape which is very spherical so that the contact area is maximized. So by that, I can lower down the overall stress between the polymeric liner and the femoral head. Femoral head also needs to be maintained so that I can get a, because if it's a ceramic, it should not break. So I should also develop something so that it becomes more tougher. So that aspect has to be also accounted for. Otherwise, any impact will break it. So entire joint will break. Again, you require some coatings and metallic stem. Metallic stem is to take that impact, and coatings again to provide bioactivity. So we have other collage of all these particular materials, and that's why all these materials are very important. But typically in a joint, we will see that mechanics is very important, because if you have a ball and a socket, your liner is again very weak. So by any movement and all that, there can be a hip dislocation because it's a snug fit. You can have an edge loading. So typically at the edges, there'll be very thin layer of polymer which now comes in contact with the ball femoral head, and that may lead to again damage as an edge loading. There can be release of wear debris that may lead to osteolysis. There can be wear of liner because that is again the softest material. Again, you can have a high contact pressure at the edges. So I'm just telling there are multiple requirements which are mandated on these hip joints, and that's what I think makes mechanics a very important part. Yes, definitely it is at a much larger length scale, but I also will like to emphasize there is also mechanics at a lower length scale related to cells and bacteria or even proteins. So if we go for, if you see the literature, there is much literature available on what material we should use. So there are various materials like polytetrafluoroethylene, polyacetal, high density polyethylene, high ultra high molecular polyethylene. But in all of them, it is shown that the ultra high molecular polyethylene is one of the best because it shows the lowest wear rate. This something out here, it is even 4 times less than a high density polyethylene. So this is the lowest one which we can ever get. So that's that becomes an automatic choice. But now we can play with the material itself by adding some reinforcement or maybe some additives, which will help us attain a different wettability. From that, I can also tailor what sort of biological response I'll get. How much of appetite if I want to deposit, then how can I do that by changing the surface free energy? Even the dispersion component of it, only by dispersion component. So if I add carbon nanotube, it starts to add to the dispersion component, and carbon being more bioactive in nature, carbon nanotube being more active in nature, it starts to encourage the deposition of appetite in the biological environment. Then again, we can play with sort of strengthening that is going to give, because essentially it is the life of this particular acetabular cup liner. So how can we bridge that, that gap of higher hardness of ceramic femoral head versus lower hardness of the liner? And then we can also see if we can also attain some added functionality. That part I can again tell, but again I can get higher bioactivity by adding zinc oxide, and also I can get good antibacterial activity via adding silver. And but that may also have some repercussion on the tribological response. So I can also see fretting or pin on this type of a wear, and that part again I will highlight later on. To again appreciate the mechanical properties of cells. So here we can see that we can get the how strongly a cell is attached to a particular surface. Because if a cell is there, if it stays globular, it means it may not like the surface. If it does not like the surface, it will remain globular. It will like to minimize the contact area. But if it starts liking the surface, then it will start to spread. And once it spreads, then it may also be attached very, very strongly. And if I take an indenter with and without the cell, so this is without the cell, this is an indenter, and then with the cell, I can see that extra energy. So this also highlights the adhesion energy that is that the cell is undergone, whether it likes the surface or not. So that part also can be compared with respect to various substrates. Again, indentation has been very widely utilized for seeing multiple aspects, even on toucan beak or hairs, how the shampoo will what is the quality of shampoo, or how the skin actually is different among Caucasians or Hispanics or anyone else, or even Asians. So that way it can provide you much more insight. So in nanomechanics, through indentation, nano scratching can provide you very high, very dramatic insights, to biological material. That's why I think this aspect is very important. Again, you can utilize atomic force microscopy. So in this case, there is something with and without the silver, and when there's internalization of silver, then also the mechanical properties will be different. So once something becomes dead, it becomes more stiffer. So that aspect also has been highlighted in various cases. Apparently, there is one very nice study by Subra Suresh. I did not actually take it up here, but generally, when we talk about cancer, the biopsy can detect it at much later stage, third or fourth stage, by the time it is already very late. But here utilize this nano indentation, and he could actually sense that cancerous cell actually they have very less elastic the stiffness, because they are continually dividing, so they have to remain elastic and they are very softer compared to a normal cell. And apparently their detection levels were pretty high, I think 80, 85 or maybe more than that, whereas biopsy has a detection level of around 30% or so. So that part actually highlighted that, how nanoindentation come into picture, and very, it can contribute very strongly even to medical sciences. So that was very nice finding. I did not cite that paper here, but I think that part, if you're interested, you can take a look. I also have done something on seeing the tricuspid heart wall. So this is the tricuspid heart wall of the rat. And I can also see how the properties change only with the pigmentation. Which means if the rat has a color of black, the heavy pigmentation shows higher stiffness. If something has a no pigmentation or a white rat, the heart wall actually will be much softer. Right? So pigmentation also actually alters the way in which the heart actually will respond, or what is the lifetime of this particular animal. So those things are also, I think, very important in getting to know how that life cycle gets affected by mechanical property of our own cells. By the way, these tests are not possible by our conventional testing. If I want to do a tensile test, I'll have to take the cell, maybe stretch it, and that becomes very tricky, right? So in this case, I can now take, and also the cell has to be live. By the time you make the sample, by the time you put it in the machine, by the time the cell is already dead. So that way, maybe the problem in conventional testing, because you also need to have proper grips, machine has to be aligned and all that. But here things can be made very rapid. The test can also be done in the body fluid itself. So that way, we can also utilize a fluid cell tip that might be available with Bruker, and that also can be utilized for undertaking some mechanical tests. So if you go by seeing the stiffness, so stiffness is pretty low for biological entities. So you can see order of kilopascals, right? And so that part I think you can see elastic modulus order of kilopascals. But when I come to materials like ceramics, polymers, even polymers have something in the order of gigapascals, 1.5, less than 1. But here you're talking about not even megapascal, you're talking about kilopascals, right? And for ceramics, it can be as high as around hundreds of gigapascals. Same thing for the metals. So there has to be a match. If I want to utilize something, I need to have a match of modulus as well as strength. I want to replace that. So that way, again, it requires some sort of tweaking. That part I think I will not get into. But I'm just mentioning that if I want to see an entire cell, I may want to go for nanoindentation, nano scratching. I want to see something of the softer cell, I may want to go for atomic force microscope. So that also I think will let us choose which technique we would like to go forward. Then it becomes very important. So in a medical setting, in a hospital setting, you will see that typically 95% of typical bio implant will not face any infection. Only say typically 5% will face infection, but that is very severe. Because revision surgery cost it's not 20 times, it's 2 to 3 times more than of a primary surgery. But pain is very high. So the person has just undergone surgery, and now person is required to take one more surgery. So it's not 20, it's 2 to 3 times. I'm saying 20 times, but that may happen because the person may be not washing their hands properly. Even when the surgery is going on, the door may open multiple number of times. And also the surgical tools themselves may not have been sterilized properly. So there might be various reasons in which the infection can actually reach the patient, or the bacteria can reach the patient. So anyway, those things can be avoided, and even when the surgery is going on, the door may open multiple number of times. That is very strange though. And typically we'll see the way in which infection will occur is there'll be planktons, and planktons will now tend to colonize. They will need to find a surface, so they can only grow on a surface. So they require some surface. They will colonize, start to form a biofilm. Once it forms a biofilm, that's it. It will become resistant to drug, because bacterial surface also has a protein which so even our natural immune cells will not be able to recognize, because that is a natural protein on the surface once it forms a biofilm. And then it starts to spread, and then once it spreads, then some of it actually may die, but some of them can also get released as a new planktons, and they can go and start spreading the infection. And that's how this step is. So first stage is called as a planktonic regime, second stage is called as the interaction regime, third region is a lethal regime. Some of the cells die. So our attention typically goes in the x-axis. We typically ignore y-axis. Y-axis very nicely says adhesion force. So if I'm within say few nanonewtons, I'm in the adhesion regime. Only when it goes above say 4 or 5 nanonewtons, then I reach the interaction regime, and then after 10 nanonewtons or so, I'm in the lethal regime. So can we utilize this? Because if I'm within this planktonic regime, at least my immune system can take care of that infection. Also, if I take any drug, then that can also help me to overcome this infection. So planktonic regime can be treated. Interaction regime actually causes infection and also spreads infection. And in typically any joint, I just listed that we have multiple entities like metal, ceramic, polymer. So apparently anything can be replaced in the body by this. So we have taken one metal, two metals like stainless steel and titanium, ceramic, hydroxyapatite, and polymer, ultra molecular weight polyethylene. And as a model, we have taken S. aureus, because we could have also taken any other bacteria, but to start with, we have taken S. aureus as the model bacteria to start with, to see its infection with all the three classes of materials. So I'm highlighting that variety of materials which are utilized in the body. And as I was mentioning, the choice of technique depends on what we want to actually test. If I want to check an entire cell, then I would like to go for nano-indentation, nano-scratching. That's what you're able to see it here. What is the force rate? But currently I wanted to look at the protein, so we have gone with atomic force microscope. And see, these are the quantitative techniques. There are other qualitative techniques which will only tell you, after say 10 wash cycles, I'm getting all the bacteria out. Only at a suction pressure of say so many millibars, I'm able to remove the cell. Or at that RPM level, I'm able to remove the cell. So these are only qualitative ways, and they may not stay valid for any other system, even with the same cells. But on the right hand side, it is quantitative, and that what actually helps us, because now it is a way in which engineers actually talk. So that's why it is very important that we go about seeing with nano scratching or atomic force microscopy or other techniques. Even cytodetacher also is more like taking an indenter and pushing it forward. So it is also more like a nano scratching, cytodetacher. There are other techniques, biomembrane or your optical tweezers, but they are much lower. So moving on kind of a smaller molecules. Right. So here we have used AFM, and in AFM we see that we have force equal to minus K into Z, or where this K is the spring constant, that is the deflection. We would like to have the maximum deflection. So intuitively we think we should keep K as low as possible, so that I can have a very high deflection. But that is not so, not so good, because if I keep the spring constant very low, then even thermal fluctuations which are there in the room temperature, that also will start giving some signals. That will mean higher background, which I do not want. So typically K is taken as typically 1 Newton per meter or so is the typical value for the spring constant. Here what we are doing, here we take a sample. We have a cantilever, then we have a tip, cantilever, and then there's a laser which shines on it on the top of the tip, and then it gives a signal on the position sensitive photo detector, right? So instead of this tip, we're replacing this tip now. Now we're replacing that with a cell, with a single bacteria. Now in the same process, we will have this cantilever. Instead of tip, now we have a cell, and now we will take this cell and now we'll bring it onto the surface, and then we'll see how strongly it is attached and how much force I need to spend for detaching it. So something like this. So I take this tipless cantilever, I bring it into a solution, and then I have a glue. Glue is strong enough, and I would only want 1 or 2 bacteria, not more than that. I just want 1 bacteria, and then now I take it and now bring it in contact with the substrate, and then I can basically see the degradation force. Now, so to see that I am getting only 1 bacteria, so I will have a solution. So there were 10⁸ bacteria in this solution of 1 ml, but that was leading to multiple number of attachment of multiple bacteria. So green one is a live bacteria, red one is the dead bacteria, but I can see there are so many bacteria which I do not want. So now what I'll do, I'll lower down the concentration. Now here I'm seeing that I only have 1 bacteria. This is a fluorescent image. So we have tested before and after the testing, and we still see there is only a single bacteria on the 1 or 2 bacteria, but we would like to get signal only from 1 bacteria. So we have taken all these materials, like which is polymer, metal, and ceramic. And here we also observe their roughnesses, which are order of 100 to 200, 300 nanometer, more or less similar, because the bacteria itself is around few micrometers. They also have the typical surface Vettability characteristic. But what is interesting is on the right hand side. Once I have this particular cantilever, I attach a bacteria on the bottom and now bring it in contact with the substrate. I keep it for 0 seconds, 5 seconds, and 10 seconds, and then we detach it. And once we're detaching it, we are seeing this response. So now there are 2 things to observe, very important things to observe. First is, for a 0 second, you will see there is a small red curve. So the force required to detach it is much lower. And as I go with increasing dwell time, so 10 seconds, this pink curve, the force values are actually much higher. Not only that, the distance or the interaction between the protein chain and the substrate also increases from 0 second to 10 seconds. That part you are able to appreciate. So distance also actually is now increasing. So you can see something here, right? So something like this here, here, and here. These are average curves, but now you can still see they are more or less increasing, and also in terms of distance, also in terms of forces. Now, second important part to also appreciate is, if I take polymer, I have some maximum level of force. For ceramic, it is higher. For metals, it is the highest, right? So this also is a difference, which is, I think, not so obvious, but the forces also differ a lot for the same duration when I talk about polymer, metal, or a ceramic. This is by this particular AFM technique. But if I go conventionally, so by conventional testing, it will show me that, oh, if I had control sample, I take other samples of polymer, stainless steel, and titanium and HAP, I will see the maximum number of dead cell in ceramic, which is hydroxyapatite. So intuitively, I will select hydroxyapatite as a biomaterial because it is antibacterial, maximum number of dead cells. There are other quantification techniques conventionally available, and that is direct contact test and an agar diffusion test, agar plate test, agar diffusion test. So in both of them, I will see that, oh, the colony forming units are minimum. It means the minimum bacterial colony is being formed in hydroxyapatite, which is ceramic. So that will be my ultimate choice. So conventional knowledge tells we should go by selecting hydroxyapatite. It ignores everything else. But now what we have gotten from our results, it shows no, polymer is showing the lowest adhesion force. Which means what? I may have a higher number of bacteria on polymeric surface, but if I wash it, they are attached so loosely that I can wash them off. So polymer, though it has higher number of bacteria on its surface, but it is very loosely attached. On hydroxyapatite, the numbers may be less, but they are little strongly attached. So you can see the maximum adhesion force is the order of 7 or so nanonewton, which means already in the interaction regime. Whereas polymer, maximum force is only 4, which is transition to the interaction regime. So they may be still in the planktonic regime for lower duration. So that also the pull-off distance also is much lower. Means that they are still, trying to interact, and they are still in that regime. Okay, so I just want to highlight the adhesion forces are very different, and for metals it is the highest, order of 10, 15 or so nanonewton. They are I think very strongly indicating that they are already in the lethal regime. It means it is ready to spread further. Infection is ready to spread further. But now we have, once we have the forces, we can transition them or delineate them into short range force and long range force. So short range force basically convey that how much of infection I can get, initiation of infection, and the long range force will convey how strong is that bond. So it's more like a chain. So how strong is that chain will be given by this FLR and the number. So I may have 10 chains. And those number of chains, what is the strength? I'll be also interested in seeing the number of those chains linked to the substrate. So that also will give me the initiation of infection, right? So because there are multiple number of proteins on the surface, some tactoic acids, so some surface proteins and all. So those aspects are being delineated with respect to the short range forces, and that's what will lead to infection. And if I just delineate that, it is a very nice observation or comparison which conveys that hydroxyapatite within 5 seconds is already in the interaction regime. So it will have a chance of very high infection. Polymer, even till 10 seconds, it is still transitioning. It is still lower than 4 or 5 nanonewton. So it is maybe still transitioning from planktonic to interaction regime. It means that still there is a chance that our body can take care of it. Whereas stainless steel or titanium, they already, I think they have very strong interaction with bacteria, and then they can directly be in the interaction in the lethal regime, and they have very high chances of infection. So it can become a little bit of a problem. So I just want to highlight this finding has proved the convention wrong, that it is hydroxyapatite which was supposed to be taken as a biomaterial for antibacterial property, but that is not so. This was very nice, I think, this belief which was proven wrong. So I will go a little fast here. I think I would like to give more time also for any discussion. So we have taken this polymer because we realize that it is becoming good. We have added some antibacterial agents like silver and also zinc oxide. Why and what, we will discuss it later on. And again, we see this profile of load displacement or adhesion force and distance, how things are being changed with respect to the attachment de-adhesion of the bacteria from polymeric surface. But now with the addition of silver, so when the silver, it is given as AAG, so 1 weight % or 3 weight %, or zinc oxide 1 weight % or 3 weight %, right? Or a combination of that. So that part we have seen, and we appreciate that silver is a more better antibacterial agent. So a combination actually is giving a very good result in terms of maximum adhesion forces, least more or less in 1A, 1Z, or 3A, 3Z, or even in 3A. So silver is much better in terms of that. And then again, we could delineate what is the frequency of forces that is happening. And again, we took out the standard or the conventional test. And that's where again we see that the 3A, 3Z and 1A, 1Z are showing the least colony forming units. So we would have anyway selected this, but we recommend that this one might be better. Why? Because of other things which we are actually showing it later. But again, you can see the silver is showing the one of the very good antibacterial efficacies. So what we've done to visualize what is happening at material level, especially for polymers, ceramics, and metals, we've taken a protein. But again, the problem with protein is which conformation to take, which protein to take. Because there are multiple number of proteins, and each protein will have, I think, millions of configurations of which configuration to take. So we have taken one particular protein, taken which is representation of that particular protein, call it as a residue, and we take its conformation which is 30 to 40% prevalent. So that's what I think makes it very appealing and also well acceptable in terms of molecular dynamics. And now we have taken this particular protein chain, and now we are trying to see what the reactions are. So once we attach it, we attach a dummy molecule, and then we stretch this in. So this is a chain. We anchor it. So this one is anchored on the substrate. This is a dummy molecule, but now we attach it or tag it to the last molecule of the chain, and then we pull it off. And then we observe how the response is. So here we have taken 3 different proteins just to also check what is the response of, say, molecular weight of the chain. So we have staphylococcal protein A, clumping factor A, and serine-rich protein A, serine-rich S protein, and they have a very different weight. You can say this is around 10 times, difference in terms of around 10 times. Also, they have difference in terms of the sex structure, also the residue count, but that part I am not getting into. And here you can see this is how the protein will look like. So staphylococcal protein A, you have a clumping factor, and then this is your serine-rich S aureus protein. They all belong to the this S aureus bacteria. And we have tried various combinations, whether the protein is lying horizontal, vertical, what sort of substrate, if I am moving the substrate, or if I am moving the protein up, or if I am moving the substrate down, and what all is actually happening for all the proteins. So that part, I think, let us not worry about that part. But what do we observe here? So I'll give two confirmations. So in this case, I have moved the chain. So once I have this protein, so this is a protein, and this is the substrate. This is silica. I can either move this protein up, or I can move this substrate down. And this looks, oh, it's the same thing, but it is not the same thing. Because, so once I move the chain up, I'm getting some forces and distances. Now, if I move the substrate down, I can see the distance actually has increased. And I can again see step-by-step change. And again, I just want to highlight that, it takes little long time to simulate. So from here to here, it might take around 7 days. I know the initial and final confirmation only. But once I complete the simulation, I see, oh, there are some intermediate points, P1, P2, P3. So what I'll do, again, I'll start here, I'll end at P2. And now I'll have starting and end confirmation, it takes around 2 to 3 days. Then again, I start at P1, and at P2, again, it takes 2 to 3 days, because it is very heavy file. The atom positioning is very heavy file. So I just get initial and final position of atom. So that's how I get this particular plot. But what I want to highlight is scheme 1 and scheme 2 are different. Because when I move the protein away and when I move the substrate down, there can be a difference in the terms the way these protein chains open up. When I move the substrate down, it starts to lock anti-parallel helices. So that's why I require a higher force. So if I'm moving the protein chain up, it is giving me scheme 1, that is a little lower adhesion force. But when I do the other way around, it is actually a scheme 2, and then it requires a little higher level of force. And anyway, it is very difficult to move the substrate as such. Anyhow, but I'm just mentioning that this is not, it looks intuitively similar, but it is not. At a molecular level, it becomes very different, because just the way the proteins open up. So now going to the second one. So I took the clumping factor. So first was serine, the staphylococcal protein A, which was very lower atomic weight of 6.8 kilodaltons. Here it is 359 residue, and it is around 5 to 6 times bigger molecule. And what do we observe? We observe the forces are more or less similar, sort of 2.53 nanonewton. I can go to confirmation and all that, but things are basically opening up. So the alpha helices, beta chain, you will see that beta chain is shown by yellow region, and by the end they all are disappearing. There can be alpha helices which are typically shown by blue, and they also tend to disappear later on. They are just opening up, and they are just disappearing. But the forces are still order of nanonewtons. And again, we took for serine-rich S aureus protein. Here also the forces are still 2.53 nanonewton. What just happened? I don't know. Am I still audible?
Yeah, we can hear you, and we can see your computer screen also.
Yeah, I'm sorry, I don't know what happened suddenly. I don't know, I'm not able to see this presentation. I don't know what just happened. It just went off.
Could be the PowerPoint itself. Sometimes it crashes off this.
So just want to highlight meanwhile that whether we take a smaller protein or a larger protein, so we want to even 6 times bigger protein chain or even 10 times bigger protein chain, it is still showing that it is not much different in terms of addition, de-addition forces. So that's what we're able to visualize using computational modeling. So I'm just telling we are doing experiments which are non-conventional using this AFM. Also, we can extend it to scratching, nano scratching. We also have done conventional, assays, and we're also doing molecular modeling. So that way, actually, it translates them to all 3 different length scales and complements. And now even if we go for this 10 times bigger chain, still we are seeing the forces are still in the region of around 2.5 to around 10. Okay, that's okay. And I just mentioned that it takes time, so we are just going back and now we're translating, and now we're able to link that our forces are order of 2.5 to 3 nanonewtons. Then what is happening at each and every location? So we can now see there were some alpha helices, some regions R4 and R5 regions which are now disappearing, or some beta sheet or alpha helices are getting destroyed. So that part you can basically see out here at what each force what might be happening. Then once we get this particular plots of different peak forces, we translate them and see now what are the long range forces using Poisson's regression and what are the short range forces. And now that will tell us what is the number of events that might be happening in that particular substrate, that particular protein. So we can see there are only 1 or 2 events might be happening in protein, but 2 to 4 events might be happening in hydroxyapatite, ceramic, and metals. It is much larger. Not only that, hydroxyapatite has the strongest long range force, strongest short range force. So it is a bioactive material. We don't just call it bioactive material. We were seeing antibacterial response doesn't mean it is not supporting biological activity, right? So that's what it is showing, that it has a less number of bacteria but very strongly attached bacteria, and others for polymer it is one of the least. So I can just wash it. So when we say should not use plastic bottles, I'll say it is okay as long as you wash it properly, bacteria will not stick. So that's what I think it was able to highlight, that the number of events and what is happening, especially for polymers. So in summary, that's what that was the deal, that we are getting very low, very high level of long range, short range forces. For polymer, it was the least. We also have done this test, I think, anyway, using it horizontal and vertical. For both of them, it was showing more or less similar response. There was not much difference. But when I move the protein up or when I move the substrate down, that is different. That is different. In real life, I can move the protein, but moving substrate will not be that easy because smaller entity will typically move. So that has a difference. And third part was regarding the material. And we could say that metals are highly, they will become more infected much easier, much faster. Add much more, but I think it is almost, I think how much time will I have?
Yeah, we have like a couple of, 5 minutes you can say, and then we can take 7 minutes for Q&A.
Okay, then I'll quickly then take it forward. So typically, so what we have done, we've taken this particular liner, zinc oxide. Now, first thing is we also assess what might be the estimation of elastic modulus, because we want to utilize it for as a liner. Then we add something, what might be the response. So that part we can utilize some mechanics and see, how this is helping in terms of either reinforcing it or, not helping it that much. Tribology is again very important. So tribology again at different length scales. So fretting is typically done for contact kind of a vibration. And there, what do we observe, that incorporation of silver is lowering the wear volume. Zinc oxide is actually not so good. It is increasing the wear volume. Why do we add zinc oxide? For increasing the bioactivity. Silver is definitely for antibacterial. So zinc oxide, by the way, zinc oxide also is an antibacterial agent, but it also is a bioactive. It also helps in bioactivity, but it is increasing the wear rate. So in fretting, zinc oxide is not good, but silver is. I go to, then we have done the whatever elemental mapping, quantifying everything, but that part I will not get into. Now we go to micro scratching, and in micro scratching again we've taken the scratch using this micro scratching, and then what do we observe here? That coefficient of friction is more or less similar, but what is again here? I see that zinc oxide actually is helping here. So addition of zinc oxide is okay. First, I should say it is decreasing the wear resistance because increasing the wear resistance, okay, that is fine. Because the wear volume is the least. So in fretting, zinc oxide was actually harmful, but here I am observing that zinc oxide actually is helping, or it is lowering the wear volume or increasing the wear resistance. That is very strange. So how one entity can increase as well as decrease somewhere else, right? So because this is the different methodology. In fretting, what is happening? We are giving some sort of a frequency or vibration. So even if the zinc oxide moves from its space, it's a rod. When it moves from its surface, then it creates a gap in between, and that gap is taken as wear volume. But it is not gone out completely. It is a harder. Zinc oxide is much harder than polymer. So once I go to a higher length scale, then I realize the real potential of it, right? So it is actually helping, not deteriorating. And again, we go to even higher length scale. So we have done this pin on disc, and pin on disc again, it brings out the real, basically real response. So here I see that 1A 1Z is coming out to be with the least coefficient of friction. That's why I was saying that we should prefer this. So if we get more antibacterial response, but it also has a higher friction with respect to the surrounding. Now here I can see that that's why we have to have. So though it is giving low wear rate, but I'll still recommend to go for 1A 1Z because it is the right amount of silver, right amount of zinc oxide. Because higher silver also is not good. It starts to lower down the cytocompatible response. So we have also seen for hydroxyperite system that higher silver content actually lowers down the cytocompatibility. So that also is not good for higher silver content. So we basically recommend that we should go for 1A 1Z because of its optimal response, either in terms of cell attachment, and bacterial antibacterial efficacy, wear and tribological response. And we also have done seeing some activity, and that's what we see, that zinc oxide is much better. It gives rise to much higher apatite deposition on the surface. And again, we have done tensile testing of various materials, and then also it tells that how we can get synergistic effect by adding silver as well as zinc oxide. So this is the summary. We would like to get basically some sort of porosity in femoral stems, scaffolds, and that part we can also add more functionality by adding zinc oxide, by adding cerium oxide, by adding carbon nanotube. And this surface response again is very important. That's what we just saw. And for polymeric liner, we can also change the morphology of zinc oxide. Right now we're using rod, but we can also tailor it to get some sort of a antibacterial response. And then the other aspect is also femoral head. That's where we can get some sort of a hydrophobicity, some texturing done on the surface to obtain appropriate tribological response. So, but I'm just highlighting, this is more or less summary of what direction we are working on. And so then summary, I'll just mention that SRS has shown addition least in polymer, which is contradictory to conventional wisdom. And again, we can see bacterial addition is decreasing with silver and zinc oxide, but there's an optimal combination of 1A 1Z to obtain this part of good tribological response, bacterial efficacy, modulus, and everything. So with this, I think stop here. I would definitely thank Professor Henny Mai and Hank from University of Groningen, and also Dr. Fahad Alam, who has mainly done this AFM work, and Mr. Arindam, Mr. Neeraj, who have performed this simulation. And I also will thank Dr. Pratyang as well as Dr. Asif for inviting me. I did not write Dr. Asif's name, but I thank both of them, and also Bruker for inviting me or giving me this keynote talk. So I'll stop here. I'm sorry for intermediate interruptions, but I hope it was able to convey what.
No, it's been absolutely a pleasure and really very insightful talk, and you beautifully explained all the techniques that are available to assess like biomechanic properties, ranging from depth sensing indentation to AFM, and then even the tribological properties. So in fact, we have multiple questions here. So first question that we have got is from Mukund. He's asking, why does wettability matter for polymeric liner?
Okay, so all these entities are in some synovial fluid. So over a joint, especially this a knee joint or any other place, so we typically have some fluid around as well. So that way it is mandated that acts as a lubricant. So if I have some fluid, then our response should be seen in a fluid ideally.
Okay. Next question we have from Deepak. Here, again, he's thanking you for the informative talk, and then he's asking, how did you ensure that single bacteria at the tip?
So for that Yeah, please.
Yeah, in fluorescent image, bacteria was not at the tip edge, he's mentioning.
Okay, so it need not be the tip edge because we bring this entire tip in contact. So as and when it starts to contact, then only I start to see the force. Otherwise, I will not see the force.
Okay, sure. Yeah, the next question is from Professor Bibu. He's asking, given the typical bacterial length and similar roughness of the substrate, wouldn't it be the case that the surface roughness will be crucial factor in addition to the surface chemistry?
If you think if I have a larger property to assess, then yes, roughness will be an issue. But here, if you're just taking few proteins, okay, in real life we will have a rough surface, and once that is there, I would like to see how the protein actually is attaching to it or detaching from it. Yes, roughness will be a critical issue. I have no doubt about that. If I am talking about larger number of bacteria, larger number of proteins, then it may be an issue, yes, because I don't know how much area I am covering. But when I am sure that I have a single bacteria, and with that level of roughness, I would like that only a few of them actually only one bacteria is coming in contact. That's a response only from one bacteria.
Sure. I mean, is also I have a follow-up.
Okay, let me put it this way. I think let me put it this way. If I have a roughness of around hundreds of nanometer, a bacteria is sitting on top of it. The bacteria will not have a roughness order of nanometers, hundreds of nanometer. It is a protein which may have. But now bacteria sitting on that surface, had the roughness been order of 3-4 micrometer, then I don't know how much bacteria is in contact with the tip, which is on the top. So once I have this particular surface, I bring the bacteria. It is basically the maximum interaction possible within that 100 nanometer regime. So that's what we want to maximize, and that's what we are actually trying to see.
Sure. I mean, he has a follow-up question. He's also curious to know the effect of substrate stiffness on the bacterial film growth. As you have shown very nicely on polymers that they have less adhesion force than ceramic. Can elastic modulus of the substrate is also a factor?
Might be. I still have a question mark. Yes, that also might be a concern, because people have seen that stiffness also will help remodeling the cells. I don't know so much about bacteria, but it feels like yes, because if I have something which is stiff or compliant, then the attachment or the way in which the molecules or proteins will interact will also be different, because proteins are governed by the conformation itself. So if the surface is able to respond to that, it may have, I think, a different, attachment. What will that be? I don't know. I am not able to comment directly, but I can see that the stiffness will play a part in terms of governing how the cellular response will be.
Okay, another question we have from Pradeep. He's asking, does mixing of silver and zinc oxide NPS affect the addition of bacteria?
Affects the
addition of bacteria.
So addition of silver and zinc oxide, it lowers down the bacterial adhesion forces.
Okay.
It does.
So yeah, last question we have from Mukund. Is the UHMWP an elastic material? If no, what are the mechanical properties of UHMWP govern the force at bond rupture even?
Okay, so polymer is it's ultra high molecular weight polyethylene. So apparently it is a thermoplastic, but apparently we are not worried about polymer. We are more worried about how bacteria is responding to that surface. So nothing is rupturing in the polymer. It is the protein chain. So if I have a bacteria, it will have some pronging proteins which will come and attach, and protein chains will have because it is not a chemical bond. So once they are detaching, that is a detachment force, more or less. It is not, chains are not breaking. It is a detachment force which we are actually trying to assess.
Okay. Okay. There's still questions coming. I think we have a minute. So there's a question for the implanted devices. How can the bacteria be removed from a polymer surface, as it is having large number of bacteria held by weak force?
Okay, no, something like this. So once you put something in the body, it is, you have to get inside the body to remove any cell or any bacteria. So we would like the implant which is getting in should not have any bacteria onto it. So for that, we are saying if I have a polymer surface, once I wash it even mildly, it will remove all the bacteria, then I can implant it.
Okay.
So I'm just mentioning that sterilization of implant is also very important. Before we put it in the body, we have to ensure that bacteria is removed. Because if you are putting hydroxyapatite, a bacteria may be strongly attached and it may not have been removed while any process, sterilization process. Then
Sure.
becomes a problem.
So yeah, I mean, I hope all questions have been answered here. But I request Professor Kantish if he stay back now, and in case if there are any questions under Q&A, will you be okay to answer them? Like you can type
actually,
in.
Okay.
If you are.
Sure. I can actually respond. I don't know.
I mean, I hope
sharing.
most of the questions are answered. But if they have any follow-up questions, they can post you. And
okay, sure. That's okay. Sure.
That's very nice of you. So with that, I thank Professor Kantish for delivering an excellent talk. So we'll go to our next talk with that.
Thank you. Then I'll take it.
Sure. So yeah, now I would like to invite our next speaker of the day, which is Professor Naveen Kumar. He's an associate professor in Indian Institute of Technology, Roper, in the Department of Mechanical Engineering. He has his PhD degree in mechanical engineering from Indian Institute of Technology, Delhi. His major research area is basically biomaterial characterization, experimental and simulation of nano and biomaterials and structures, active vibration control and fault diagnosis, and flexible and stretchable devices. He has also been associated with the Stevens Institute of Technology as a research scientist. So with that, without further ado, I would like to invite Professor Naveen for his talk now. So Professor Naveen, the stage is yours.
Thank you, Pratyank, and your team, and also Dr. Asif is here. So it is my pleasure to listen to him also. He has a talk, and I am eagerly waiting for his talk on the soft material, and which will be a very fascinating new area for in particularly those people who are working in biological materials. So I will share my screen. So here, as per my previous professor, Balani has talked about the biomaterial, and he has put the foundation of the bio side here. So I will build up from there, and the only difference is here that I will talk about the mostly the biological materials, which is we all have, or God has given us. There is no need to make. So how that the property of these materials are there, and not so much efforts has been made to get the know how they function and how they behave under different loading conditions. So we will talk about most of the time here. So, and we will limit my talk to bone quality, which is a new terms come up in when we talk about the bone materials, and we definitely talk about up to the nanoscale mechanical properties, and I will cover only their very old system and the founder of this hesitron, the Nanoindentation. Most of them my work will be focusing on the Nanoindentation. We have used it like anything, and we will show you how different way you can use it. That's a beautiful machine, and that's the beauty of the science which is incorporated in that machine. So our body is as a locomotion, and we have all these bones structure. They move, they remain stationary, and they interact each other. And how this fundamentally coming up the mechanics, to understand the mechanics. This is a beautiful campus of our IIT, which come up in about 35 kilometers away from the Chandigarh. So this is the department here, our department, mechanical engineering, and biomedical engineering is this department. So, so here, this is our department, right? Now, why we need to study these things? What at all make us fascinating and why the bone or tissue are needed to study. So you see here, the answer is coming up. So these pyramids are population pyramids, and the first two pyramids are Indian population pyramid. First pyramid is showing the Indian population at present or in 2020. So near about 30%, 31% of our population was older population, means that has crossed 45 or 50 years of age. But if we talk about in 2050, nearly 46% of our population will be older population. So that's why we are saying our country is a young country, but it will not remain as young, and it will, after some year it will become the older country, and it will be similar to what at present the USA has nearly 48% older population, and it will further increase to 52% by year 2050. So once the population become older, they have the major issue related to musculoskeletal system. All those bone and tissue related disease come up, pathological conditions come up, and then that's why the understanding of that is not only for the clinician surgeon, for engineering point of view is also very important. I will show how the engineer point of view is very concerned and very much needed, and which all these clinicians and surgeons are also need that input from the engineers. So now, our body has all this loading condition. We also come across the static loading condition, where we are exerting the constant amount of force, but also we have come across the dynamic loading condition, that we are coming across the variable amount of load upon these, our structure, our bones and tissues. So which make it very necessary things to study it, because under different loading conditions they behave differently, because these are the natural materials as well as they are viscoelastic materials, so they are load dependent, frequency dependent properties are very important to study, and how they behave. So under loading condition, if we talk about the femur bone, this is a femur bone, which is the longest bone in our body, and it is this is a femur head, right? So, and this is a femur neck. So when we have talking about the static load, so load come up here, and then we have these all type of stresses, the compressive stresses they come in the medial region, and the tensile stresses come in the parietal region. Similarly, at the neck, that happens. The compressive stresses are there, the tensile stresses are there, and most of the time during the fall or slip conditions, the failure happen here in the neck, and neck failure happen where the fracture happen in the tibia. Now, under dynamic loading conditions, again, we have the beautiful zones. Our bone has the capability to take care of the compressive load, tensile loads, and those zones are there in our bone, and they are so this tabiculi or the fiber of these bones are aligned in such a way that they take care of this the different type of loading conditions which occur during the static or dynamic loading conditions. So that is also a very beautiful and fascinating thing to see, that how they take care of the compressive and dynamic load. So generally the fracture happen when you come across the abnormal loading directions, and or which most of the time the fall conditions, and here this the most of the time the failure happen at the neck region. So how the bone fail, that's also a very interesting phenomena to see that. So what happening is, when we have this we are applying the load, some deformation or elastic within the elastic limit, if we are within the elastic zone, we are there, then there the failure is happening, like we are showing in the upper figure. So these are the a typical cross section of the bone. We are showing a bone cell here. This is the osteon, and on the periphery the matrix is there, right? So when this is our load is crossing the limit, then we have this micro crack initiation happen in the bone. So you can see the micro cracks are initiated, and these cracks get propagated, and which is happening in the plastic load. So these cracks propagate, and then structural failure happen, and then these bones failure happen, and then you can see this not only the matrix, and then our bone also has this the collagen, the proteins, which our earlier professor has talked about. So they are embedded in the bone, and they make it this collagen fibers are proteins, and they are also contributing in the stopping this micro cracks or the failure. But if we are crossing some certain limit of the load, then definitely the failure happen in the bone as well. Right. So there are two major things which we try to study. Most of the engineering materials, as well as here in biological materials also, the material or structural behavior. So the material property, we generally talk about this to investigate all the material property. We have small samples here, like a bone biopsy, or during the surgery, we get some samples and try to investigate the material property. But to get the structural property, we need the whole bone, or a handsome amount of the bone length, and then we can find out the structural property. Here we will talk about only the material property of the bone, which we have investigated by using this most of the time the nanoindentation technique. So now, how this bone quality come in the picture? So now I'm connecting a little bit with the clinicians. These clinicians, most of the time, this orthopedic surgeon has one tool, which is known as DEXA, which is a X-ray based technique, imaging techniques, which give the bone density. And based on this bone density, these clinicians try to categorize the bone, whether bone is having a good strength or bone is weak. That's the only tool which they have. But over the years, clinicians also feel that there is something else is needed. The DEXA is not able to clearly distinguish the different types of bone, because they have seen that bone seems to be good, but this DEXA is giving not good results, means saying that bone is not good. The strength of the bone is low, but whereas they find that bone can survive for longer time duration. So this based on the DEXA, they give the medication. So then these new terms come up, the bone quality, which was lagging in the BMD, which is still clinician has only one tool. So our work is more related to how to incorporate some more factor in the bone quality. And if we add those factor, then can we talk about the fragility factor, the chances of the fracture, which happen if we have a minor trauma cases, or the side fall, or major trauma cases like accident, or the fractures happen. So this bone quality, what is this bone quality? So what we are saying that two people may have the same DEXA score or bone mineral density, but the chances of bone fracture at low impact will be different. So how that's happen? If they have the same BMD, they should have the chances of bone fracture similar, but that's not happening. So that's why researchers are digging more. What else is there in the bone? So these are some of the factors. So National Institute of Health come up with the term the bone quality, which is talking about the sum total characteristic of bone that influence the bone resistance to the fracture. Now, what are those characteristic features? Which features which we come out in our recent paper, JCM paper, Journal of Clinical Endocrinology and Metabolism. We talk about these are the factors. If we incorporate this more factor in addition to BMD or the DEXA score, we can come out a very good estimate of the bone strength. So what are these factors? Major factor is the mechanical and material properties, protein content and protein secondary structures, bone mineral crystallinity, more mineral content and mean crystal size. The bone microstructure is also contributing. bone micro damages, I have talked about a little bit in my previous slide, bone cell activity and dynamics, and then accumulated advanced glycation end product, which happen in some of the cases where the pathological condition arises. That case is the diabetic case. This AGEs get accumulated in the bone and which deteriorate the bone property. So if these 7-8 factors, if we include, and in addition to DEXA can be a screening tool, but if we have the proper knowledge or proper values of these parameters, we can estimate the bone quality more easily, and bone strength can be ultimately can be determined. So here in this talk, we talk about the material property, how we can find out the material property. We have find the other parameters, but because this is not the forum for the other parameters, the audience who are interested to see the other parameters, they can see our paper, JCM paper, and we have talked about the other parameter also. Now coming to the hierarchical structure of the bone. So we have this full bone, the tibia bone or femur bone. So at the mesoscale, we have this distinguish between the cortical bone. Cortical bone is the outer peri bone, which is a little bit more harder bone and denser bone than the trabecular bone. Trabecular bone is the inside bone, which is the little bit spongy bone. And then if we talk about the little bit more deeper scale, which is from 5 micrometer to 500 micrometer. So we can talk about the microstructures. So we have this osteon level, we can come up. And then in case of the cortical bone, and we can come up with the trabeculi in case of the trabecular bone or the inner bone or the spongy bone. And if we talk about the nanostructure, then you can have these proteins, the minerals, crystals are there. So the bone is made up of the proteins, the minerals, and the biological waters. So they are these combinations of them are they come up at the picture in these at this scale, at the nanoscale. So bone has about 50 to 70% of the minerals, 20 to 40% of the matrix, and 5 to 10% of the water they have. Now coming to different techniques, there are so many techniques are there, which we can get the property at this length scales. Like we can have the macrostructure, whole bone testing, different equipment like UTM you can use, or fatigue testing, or different mechanical testing are there. Then DEXA is also QCT or PQCT, which is also for structural properties you can use. And then coming to mesoscale, the BMD, the TBS score, fracture risk vertebral body for vertebral columns. And if we talk about the microscale, the micro-CT, the SEM, the histological, reference point indentation. So many techniques are there, and you can get some of these parameters, right? And our nanoindentation also come in this, microstructure properties or material properties we can find out. And if we talk about the nanostructure, then this FTI, Raman, AFM, and so many other parameters are there. And nanoindentation also come in this range. That's depending on what type of tool or tips you are using. So here in our lab, our so many years, this we have TI 950. I think it's about 7-8 years we are using it, and our lab name is Hasotum lab, which have the hard and soft tissue mechanics lab. Now, as most of the you were knowing this, that how we get it. So we are applying a load. We have the load cell in the machine, which can measure the load even up to a few micronewtons, and we can measure the depth in nanometers. And then even the load cell we have now to measure this in the nanonewton forces also. So we have this load displacement curve. So during the indentation, the holding, and then taking out the tool, and from there you can find out different parameters, particularly the hardness, and you can find out the modulus here also. So some relations are there. These relations are used to find out those parameters. And once you find out the reduced modulus, you can have this the Young modulus of these materials, which can be find out by using these relations. These are the cited by many researchers, and these are the now well-established relations. Right. Now coming to how we have done this, use this indentation technique. So here what happening is, in case of fragile fracture, we have this femur fracture happen. And most of the time this neck, the fracture happen at the neck. So, and these, as my previous professor talk about this, the implant. So this surgeon has to put the implant, they have to take out this femur neck and head, and they discard it. Either they keep it in the bone bank, or they throw it in the dustbin. So we have the ethical approval to collect these samples. So we are collecting these samples from this PGI MER Chandigarh hospital. So then what we do, we are cutting these samples by using the low speed sectioning saw. And here you can see this lateral and medial portion we have. And so here we have this cortical bone, and this is the inner is the trabecular bone. So this is the spongy bone, and the outer bone is the more denser bone. And these collected bone are then embedded in the epoxy, and we make it smooth surface. We have these surface finishing tools. We'll show you maybe in the next slide. And then using this PI 950, they have these transducers, different transducers and different tips, appropriate tips you can use it. So we are applying the loading condition and unloading condition, and you can get this low displacement curve. That's how this we are doing this sample preparation and experimentation. Now, microstructure of this cortical bone. The cortical bone, which I have talked about, this is the outer peripheral bone, which is more denser, and trabecular bone is the inner peripheral bone, which is a softer bone. Right. So you can see this on the femur head. This you can see this. This is the outer periphery. This is the cortical bone, which is very thin, having very few millimeter on, even fraction of millimeter sometimes. So here we have this slow speed saw, and we have this polishing tool. So here, if we say this polished our cortical bones, so here we have this osteons, the bone cell, and the matrix, and then this is a single tabiculi in the cortical trabecular bone, right? So that's how this microstructure look like. And we put these different indentation at different locations, right? So these are the marked location, and this is a typical indent which look like here. Now, this is the case we have talked about this type 2 diabetes. So what is happening? We are trying to see that how this bone property change, material property change in case of the patient who has type 2 diabetes and whose HbA1c level is quite high. So there is a threshold parameters, like 6 is the threshold parameters of this glucose level. If it is more than that, it is considered as diabetic. So based on that, we have this the number of samples, collected number of samples, and we try to find out this the major parameters, that modulus and the hardness, by using this nanoindentation. And we find you can see that non-diabetic and diabetic, and then we try to correlate. So significantly correlation we got in both the hardness and modulus. It means that once the patient has diabetes, then this bone property get deteriorate. You can see the modulus is get reduced, as well as the hardness get reduced for these patients. So that's come out from this study. Similarly, we have seen the osteoporosis, how this is happening. So this very interesting finding. So here we are showing the BMD, which clinicians have this through the DEXA, which I have talked about. So you can see DEXA, these patients are having the about same value of DEXA, means the bone mineral density, which DEXA give is about same for osteoporosis patient and osteoporosis with type 2 diabetes patient. It means DEXA is not able to distinguish these two different categories of the pathological condition. It means we need something else, and which this surgeon and clinicians was talking about, that there is something else because they find that bone is not so deteriorate, but DEXA is not able to tell us. So we have done this indentation and find out the hardness. You can see that osteoporosis has, but if osteoporosis plus type 2 diabetes, which generally happen because osteoporosis is related to age, and type 2 diabetes also most of the time happen at the older age. So if we have this both these cases, then this the hardness get reduced significantly, as well as the modulus get reduced significantly, which is not conveyed by the DEXA. So it means something was lagging, which NIH also come out, which one of the parameters that material properties, bone material properties, it is coming out from here. So here it is clearly showing that if we are using this tool, we can further dig up and see that how these patient are having the bone strength, and what are other parameters which are contributing to the defining the bone quality. So it means material property of the bone is significantly contributing to bone quality. Then very interesting study we have done. We have again taken the type 2 diabetic cases, and the patient with whom we have collected the bone samples, we have also collected the fingernail. Why we have collected? Because this again come out from the clinicians that nail also get deteriorate. Can nail be also a screening tool in place of the plasma or the blood? Can in place of the serum, can we get this the estimate of the diabetes by using this nail? And based on that, we have seen, and what is the correlation between this the bone deterioration and the nail deterioration, because they have their own cycle. Means this bone has also a period of the bone formation and bone loss, which keep on happening in our body. All the bones keep on making, and some bones are get eaten up by the cells, and new bone formations are keep on happening. And similarly, this happening with the nail plate. So we try to correlate and what is relation, and also there is a possibility to have a screening tool like nail can be a screening tool. So this is the correlation we are talking about, the bone and fingernail respectively. We find clear correlation between the bone and nail deterioration. Means nail also deteriorate in case of the type 2 diabetes, and that is also related with the bone regulation. Means maybe we may not need the biopsy. By even collecting the nails, you can have some estimation of the bone loss, how much happening in case of diabetes. That also can come out, as well as another in another paper we have come out that it can be a screening tool. The nail can be a screening tool for the type 2 diabetes, but you have to investigate a lot of parameters, not only this nanoindentation parameter. Other parameters we have also investigated, which is there in our paper. But here I'm talking about only the nanoindentation, right? So now the major challenge is that once we are collecting this the bone samples, these bone get dehydrated, and how this till we bring it in the lab and perform the experiments, because suppose we have to perform the nanoindentation, then also this bone property change. So the environment and the water content also play a significant role. So we try to see that how this play a significant role. We have what we have done. We have taken the hydrated bone, stored in the previous soak gauge, and at the -20 degrees centigrade every step of preparation. And dehydrated bone we have not placed in the previous so gauged, but we have stored in 5 degree every 5 step and centrifugation for 48 hours. And here, what we have done is we have done this location indentation at different location at the from the endostereum or to the peristeum locations, and the number of indent, nearly 20 indents we have done, quasi-static and dynamic loading conditions. So same formulation we have adopted here. So here you can see that this dehydrated is the red one. So sorry, the dehydrated is the red one, and the hydrated is the green one. So you can see that the once bone get dehydrated, this modulus and hardness increases, which is quite evident because some of this the water loss happening, as well as this hardness modulus, as well as this because say viscoelastic properties, so frequency dependent properties also changes in case of the dehydrated bone, and which is very important to keeping in mind that the bone should not be dehydrated. From this study, we can say that when we preserve this bone or when we do this the experiment, the humidity or the water content in the sample play a major role, and researchers should be careful about. So that's some of the conclusion which we have talked about. And then some of the material properties like the viscoelastic behavior of cadaveric bone, different peak load and elastic modulus, how they are contributing. If we talk about the different peak load and the whole time which is contributing in the viscoelastic behavior. So we have taken the 5 osteoporotic cadaveric tibia bone, and we have applied these some protocol. Like in first case, we have taken the maximum load 1000 micronewton, and unloading rate is 100 micronewton per second. Hold time is 10 seconds, and we vary these parameters for different conditions for these 5 osteoporotic bone. We have taken a number of samples from these 5 bones, and then we try to place this parameter. We try to understand how this peak load, loading unloading parameters whole time contribute differently for this bone, which is important for the study of the viscoelastic nature. So here, okay, my time is, I think, I have already crossed the time. So so here we are talking about the whole time, peak time, loading rate. Priyanka, I think I am crossing my time, right?
Yeah, I mean, it's okay. Maybe a minute. Yeah.
Quickly finishing it. So I am coming to important. If I have some important, I can come up. Yes, one more study which is done by one of my students who is there in. So here what we are trying to see, you can also demineralize the bone and see how the protein is contributing in the bone, because to understand the mechanics of this the protein and the minerals separately, because what happening is in different pathological conditions these constituents change, and but in normal bone you cannot see how this constituent varies. So what we have done in this case, we have demineralized the bone by treating some of the chemicals and try to see that how this is affecting. So some of this behavior is coming out. So because these collagen fibrils and collagen fibers are there, which is at the higher scale, and if we go deep in the scale, then collagen fibrils are contributing. So we try to see that how this the proteins is contributing in the strength and this modulus based on this experiments. So that's the talk about. And then also soft tissue, you can see this the aortic wall samples we have embedded and then try to see this behavior, and we got significant things in this also. So in the soft tissue. So that's I think the process will also talk about in soft tissue. And these are all the papers which we have published. So you see, can publish excellent paper by using these machines, and it's not only our paper, but also this lot of clinicians are there. All these are the surgeon or clinicians which are helping us in this study. So this all are paper based on this single machine, the Nanoindentation. Yes, we have used these other machines, but major contribution is coming from this machine, and these are the some of my students who are contributing, helping us in this study. But maybe new one student has not added in this. Some new student also contribute. And some of my funding agencies like DST, DRDO, DBT, AOTA, and some of my collaborators also are helping us. Thank you. Thanks for time. I am sorry I have taken overshoot the time. I just realized.
Oh, it's absolutely fine. It's pretty good talk. I mean, good to know how this application can even be extended to, medical research. Like you're collaborating with the, to quantify the quality of bones with respect to diabetes, right? So it's pretty nice application that you have shown. I'm just looking for any questions from the audience. You can post your questions in Q&A, or any questions from the panel.
Pratyang, I have a question. And
Sure.
It's a very interesting talk, Professor Naveen. I think, can you highlight the importance of surface preparation for bone is such a material that it's anisotropic. What are the conditions that you prepare the samples? How do you prepare them?
Yeah, so very good
polish.
question, and a little bit I have talked about it. So that's the moisture content, that dehydration is a very major concern when you are transporting this bone. Because if you have the hospital in your institute, that's good, because very quickly you can bring it. But during during this experiment, like indentation also, loss of water may happen. So the preparation is as usual, but yes, the slow speed saw should be there, because then you are not altering the stresses in the sample, or not incorporating the stresses, the residual stresses which may come up because of the fast cutting. So slow cutting should be there, and polishing, slow polishing again should be there of these samples. So you should keep in mind, or hand polishing. You have a lot of now polishing materials are there, you can use it. And then coming to the humidity, yes, you should have maybe a incorporated separate chamber, or you can spray, keep on spraying the sample. So those are some of the very important things which we are incorporating those.
Very good, thank you.
Yeah, I see there's some appreciation of the talk. And then they were asking like whether it can be shared. So, I mean, we will be sharing the request. I hope it's okay, right, person? I mean, we can.
Yeah, no problem.
Share
I will. I think some more slide was there. I will take out those slides, and you can share that. I will share with you,
okay,
and then you can share it.
Sure.
Those who are interested, please.
Absolutely. Great. So, with that, I think I'd like to thank Professor Naveen for some really interesting talk, and you always come up with some really interesting applications with nanoindentation. That's really good. Yeah. We do remember your talk with respect to dragonfly on the wings.
Yeah, that's another aspect which I have not covered it. That's also very fascinating.
It's always great to hear you.
Yes.
Great. So with that, I'll move to our next talk. Thanks, Professor Naveen. So I'll go to our next talk. Okay, so now I would like to invite Dr. Syed Asif. He's a director of R&D at Industron Nanotechnology Private Limited. He's based in Minneapolis in USA, and he's graduated from University of Oxford with his PhD degree in material science. I mean, he's a very well-known name in the community. He has 30+ years of his experience in designing and developing nanomechanical test instruments. In fact, he has led most of the development of the instrument that we are using today now. I mean, in today's time, with respect to nanomechanical test instruments at Bruker Nano Surfaces, earlier Hydrotron. He has over 120 publications and 22 active patents, and 4 R&D 100 awards under his belt. So, I mean, it's really always great to have him here, and listen, it's always a great learning. So, without further delay, I would like to pass on to Dr. Syed Asif now for his talk.
Thank you, Pratyang, for the nice introduction. It is always a pleasure to come over here and talk about what we do. Let me try to share my session, my screen. Just give me a second. I think now you can see my screen, I believe. Okay.
Yeah, we can see it.
Okay, you can see my screen and everybody can hear my. Okay.
Yeah.
That's good. So without delay, I think I will get going. First of all, for the audience, thank you for attending this symposium. And so in my talk, what I am going to cover is about more of a technology point of view. Like Pratyang mentioned, I'm a material scientist, but I spend a lot of my life, most of my life basically, on developing instrumentation that helps to characterize materials. So it's a kind of interesting thing for me that having instruments developed for applications. Okay. So my talk will reflect that. I'll be dwelling more on the instrumentation aspect, but I'll also try to cover things that is relevant to biomaterials. So the way I'm kind of dividing my talk is kind of first I will talk about hard materials. The two speakers, Professor Kantish, Balani, and Naveen Kumar, they gave a pretty good introduction on the importance of characterization and the tools that are available for mechanical characterization of biomaterials. So I don't have to kind of give that introduction. So I will talk about the basic fundamental instrumentation that we use to assess these properties. Start with the hard materials, and then I will jump on to soft materials and some of the issues that we have to kind of still resolve in understanding the soft materials, right? It's like 25 years ago when I was a PhD student. At that time, Nanoindentation was very new and exciting. We were developing techniques and models and things like that, and then we did a lot of steps. But then recently, I kind of picked up similar enthusiasm by working on soft materials. Soft materials are still evolving. The fundamental understanding as well as modeling aspect is still going on. And also the instrumentation development. There is a lot we need to do and understand the soft materials as compared to hard materials. So I will cover a little bit of that in my talk. Okay. So we are talking about all mechanical properties. For most of you, I think you are already familiar with what is tensile strength and elastic modulus. We normally do tensile tests, right? So, but our speakers, the previous speakers, correctly mentioned, like when you deal with
Dr. Asif, sorry to
yes,
interrupt. I mean, you have, I think, your toolbar, Zoom toolbar on top of your presentation, I think.
is it? It's kind of blocking something.
Yeah, it's just blocking. I mean, if it comes out easy, it's okay.
Yeah, that's fine. I'm kind of bringing it down. I don't know how that is coming. How do I get it out there?
Normally should go off. Otherwise, it's okay.
It's
It's
not.
not blocking
Okay.
major. Yeah.
Okay. Okay. So, okay. Yeah, so mechanical characterization, I think we are very familiar with how we do with the tensile test, and but when we want to do something on biomaterials, often it is difficult sometimes, and with the depth sensing nanomechanical testing that we developed for hard materials like metals many years ago, people started using for characterizing biomaterials. It's kind of interesting. The understandings that we developed in terms of modeling the contact mechanics and all of that for metals is still kind of useful for biomaterials. And so with that, we can kind of infer what is the harness of the material and models of the material, like Professor Naveen and Professor Kantish Balani presented in their talk. So, but the advantages with indentation is that we can go down in length scale because we can use smaller and smaller probes to probe smaller areas, and we can also automate the whole thing. For example, and if you have a small sample, we can do a number of tests and everything automatically without doing any. We don't have to do manually. We can do everything automatically. Okay, that's the big advantage. And along with the advantage, also when we try to generalize our methodology, sometimes it doesn't work. So we need to also develop, for example, if you have a porous material, you can't probably use the same models and methodology that you used on the metal sample. So you need to be, you have to take care of that. Okay. So with that, let me, I think my next slide is kind of showing the idea of what indentation means. This is for people who are even new to this field. I just wanted to give you an idea. So what we do in nanoindentation is basically you take a very sharp probe made of diamond, and you apply a load through the probe onto the sample surface, and like here, and then you penetrate the sample surface, you load and unload, and you created an indentation on the sample surface. On the left side, you saw that you get a direct output of the load versus displacement. And then you can also see the deformation happening. And this experiment was done inside a scanning electron microscope. So that's why you were able to see what's happening here. When you get this load displacement curve, you can analyze the curve. For example, the unloading curve you can analyze by measuring the stiffness. If you take the load divided by the depth, that gives you the stiffness. So you fit the unloading curve and measure the stiffness. And once you have the stiffness measured, then you can kind of, from that, you can always measure the hardness and modulus by modeling. Okay, so and so to apply this force, so how do we do this? We need to generate force, and sometimes, like for example, in the airform, you can have a spring, and deflection of the spring can be measured as force. But when you need higher forces, you need to generate the force. So the way we generate with our instrumentation is through electrostatic force actuation. That is what we do is basically we have a sensor, a capaciting sensor, which has a 3-plate capacitance structure, and the center plate is kind of suspended by a spring. Now, if you apply a voltage between the center plate and the bottom plate, you generate electrostatic force. Similar way that you take a comb and you just rub it on your hair, you generate that high voltage, and then you bring it closer to a paper. The paper will jump to the comb. The same way, you generate a high voltage between the center plate and the bottom plate. Then the center plate will get attracted to the bottom plate. In that way, you can generate force. Okay, so this can be simply represented by this simple equation where force is a constant times voltage square. So simply by knowing what is the voltage that you are applying, you can measure the force. So that's how we generate the force. Now, because this center plate is kind of suspended between two outer plates, you can measure the capacitance. Now it's kind of a parallel plate capacitance structure. So you measure the capacitance as the center plate moves. The gap between the center plate and the outer plate will change. Then you can measure the change in capacitance as the gap changes. That capacitance has been changing, capacitance has been converted into a displacement. So with this simple transducer structure, you can apply force, and you can do it at very, very small forces. For example, in a nanonewton regime, you can apply forces, and then you can also measure displacement down to like less than a nanometer. So this is what we use typically for applying force and measuring the displacement with respect to nanoindentation. Okay. So in the next slide, let me see. Okay. Now, if you take this transducer now and combine it with a 3-axis piezo scanner, for example, these scanners are made of tube piezos, quadrant piezos, and somehow like these transducers are like about ½ an inch in size. You can mechanically couple to these piezos, and if you apply a voltage onto these piezos in X and Y direction, you can move those transducers basically in X and Y direction as well as in Z direction. Okay, that helps to, if you wanted to place indent at a specific location, you can do that. Or if you just wanted to scan on the surface and create a
Your slides are frozen, I think.
Now it's moving.
No, it's not moving.
Oh, I mean, it is moving on my side. Is that moving at all? Let me restart again. Okay, sorry.
Now we can see your screen. You can put in slideshow.
Now I'm sharing now.
We are not seeing anything.
Huh. Hello?
Yeah, Dr. Asif, it's been blocked. Can you put on the slideshow?
It's on the slideshow now.
It's coming now, but you have, I think, Zoom toolbar or something which is blocking at the bottom now.
Yeah, I think I don't know how to get rid of that. Do you know how to do this?
Yeah, it is okay now. It is okay.
Okay. I don't know how to get rid of this toolbar. Okay, I think we'll manage with what we have.
It is okay now. You can go ahead. Yeah.
Okay. Now you can see this.
Yeah, we can.
I'm sorry,
we can see. No problem.
Okay. Yeah. So I was just talking about like how we can combine the transducer with the piezo scanner, which can be used to scan on the surface and generate a three-dimensional topography. Which can be kind of an eye that you can use to see what is on the surface. And I'm kind of showing a few pictures here, basically, which is a scanned image with this transducer on the scanner on a wood cell, and you can see a clear wood cell here, and also along the periphery of the wood cell you see tiny indents. So this instrument not only can be used for doing inundation experiment, but also can generate a scanning probe microscopy image, which you can use to aid and understand the surface and then place indents where you want to do the indent and get mechanical properties. So you have this inbuilt capability of in-situ SPM imaging along with the nanoindentation. So that's kind of a very useful for biological materials. Okay, so that's the message that I wanted to kind of tell. And now I think I already covered that you can use this transducer to generate low displacement curve. And these low displacement curves are unique to the specimen material that we use. We call it mechanical fingerprint. And from the unloading curve, you can measure the stiffness. And from knowing the stiffness and the area of the tip by calibration process, you can calculate the reduced modulus and hardness of the material. By knowing what the load you are applying and the stiffness that you measure for a given load, and what is the depth of indent and all of that. Okay, so if you have this low displacement curve, then you can do this hardness and modulus measurement. Like I said, it depends upon the material that you're studying. Some materials are some more of elastic behavior, some material will have more elastoplastic behavior. So depending upon the material, it will have a unique fingerprint. And that's how we can use this technique to identify failure in materials, structure property correlation, and things like that. Okay, so now with respect to biological materials, we can use this nanoindentation technique, and depending upon what the length scale that you want to study. For example, let's say you have a tissue, and normally with a bigger probe, you can go and use a nanoindentation to understand the tissue property, because it's kind of measures the average property over a surface area. But if you wanted to go to cellular level, we can go even smaller probes and understand when you apply a stress through this probe onto the cell, how the cell reacts or how the mechanics happens, and then we can measure that force displacement response curve and interpret all of that. But if you wanted to go beyond that, you need to go and use some AFM-based technology, because the forces that you require are much lower than nanonewton. Sometimes you have to go into piconewton length scale, about like less than 0.1 nanometers, so picometers basically. So in that case, you can use AFM-based technology to understand and develop biomechanics understanding of the material, right? So it's all depending upon the length scale that you want to do, and either you can have different type of instruments, a microindentation, a nanoindentation, or AFM, or you can have different type of probes, a spherical probe, or a bigger probe, or a flat probe. So you have this ability to change, and you can make these measurements locally where you are trying to understand. So that is the advantage of this kind of technique. So in my talk, my title is like hybrid testing technique. What I mean to say is basically, along with this nanomechanical or micromechanical testing technique using this probe, indentation probe technique, you can also combine with mechanical something else. For example, like I mentioned about in-situ SPM imaging. So if you can combine the in-situ SPM imaging with the harness measurement using indentation, I will call it as kind of hybrid technique. Sometimes you can combine with, for example, Raman. For example, in one of my example, I'm going to go present how you can use Raman microscope and the ability to use the Raman microscope to identify chemistry of the surface, and then couple it with a mechanical property measurement. How you can do that? Okay, that's why I'm calling it as a hybrid technique. There are other things that you can do with this transducer. For example, instead of applying a force quasi-statically, you can also apply force dynamically. But what I mean is that you can apply a oscillatory force and then measure the response. We call it as DMA. So in that way, like you can have a kind of hybrid techniques in combination with this standard quasi-static nanoindentation technique to develop a further understanding of the stresses and strain that is being induced, or you putting a stress and strain onto the sample, how the sample is going to react. We can all do that, okay? So this is what I mentioned about Nanodma technique, where instead of just applying a force and displacement, quasi-static force and measuring the displacement, you can also apply a dynamic force and then measure the response. For example, if you apply an oscillatory force and measure the oscillatory displacement response, you can, if the material is elastic, you will not see a phase shift between the force and the displacement, and the force that you are applying will be in phase with the displacement that you measure. But if you do it on a viscoelastic material, there will be a delay between the force that you apply and the displacement that you measure. That will induce kind of a shift in phase. That phase shift can be measured using an instrument called lock-in amplifier. And measuring this phase shift along with the known force, the oscillatory force that you are applying and the oscillatory displacement that you are measuring, you can measure the viscoelastic characteristic of the material, namely the loss modulus and storage modulus, tan delta, and many things. Okay, so I'm not going to go through all these details in modeling. I'm just going to give you an idea that you have, along with the quasi-static force displacement measurement, you can also do a dynamic measurement in measuring the loss and storage modulus and tan delta on viscoelastic materials. Okay, so coming to indentation of hard biomaterials, I thought of giving a good example of bone, but Professor Naveen, we covered pretty much of the indentation behavior of bone. So I'm going to skip on that slide because it's kind of repeating the same thing that what he was talking to us, how you can use the nanoindentation technique to measure the property of the bone. One thing that I wanted to stress is basically bone is kind of a hierarchical structure and it is totally anisotrophic. So if you measure the properties, you will measure the properties depending upon the loading direction that you use. So one thing that I would advise is basically when you do this bone kind of sample, if you do a polishing on the 3 direction, the way that I'm showing over here, the bone material is kind of being polished on 3 different directions. And then if you make a measurement now, for example, on the transverse plane and then the frontal plane or the lateral plane, you can do this measurement at 3 different directions and you can measure the modulus. Okay, so that will give you a better understanding of how this anisotropic properties are changing. How the mechanical properties are changing on different directions. For example, if you look at this table, you're measuring the modulus on 3 different directions. But you can see that at transverse plane, the modulus is much higher compared to the lateral plane. Which shows that the load carrying capacity along the direction is much better compared to the other direction. So that gives an indication basically how our bone has been designed to take the load and why sometimes this anisotropy is being measured. And one of the reasons that we have this anisotropic behavior is mainly because of the mineral content. This is what I'm going to go and show in this particular slide, where this has been done for a rack tooth. What we have done is basically you take a tooth material and cut and polish it and put it on nanoindentation. This is a typical 950 or 980 nanoindentation. But along with this nanoindentation, if you look at in this arrangement, you have a low load transducer, and then you have a high load transducer, then you have an optical microscope. In that optical microscope, you can combine Raman so that we can use it as a Raman microscope. Now, along with this mechanical measurement, if you bring the sample under this Raman microscope, we can measure the Raman peak intensities. For example, if you have a hydroxyapatite, you will see a peak at about 962 per centimeter. So if you look at this peak, and you know this is due to hydroxyapatite, and if you measure the intensity of this peak, that gives you the mineral content. Now, we have a way of understanding the chemistry of the surface. What is that? If you have a high mineral content, can you correlate to the mechanical property? If you do that, for example, and you go and map with the Raman and then measure the intensity at that particular peak, and then you can go on indent at the same location and to try to correlate that if you have a high mineral content, do you get a higher mechanical property? For example, in this case, it's just a modulus. So you can see that readily, like when you have a high peak intensity, when you have a high mineral content, the modulus also goes up. So in that way that you can combine now Raman along with this mechanical measurement that allows you to understand in a better way of why you have this anisotropic property, why you have the modulus of the material or hardness of material at some location much higher compared to other locations. So you have other kind of tools that will enable you to understand in a better way. Okay. So I wasted some time, so I'm going to go here and increase the speed of my talk. So instead of now giving more on hard materials, I'm going to jump on to soft materials. And soft materials, the way that I am kind of classifying as soft is like anything. If you have a measure, if you take the modulus, and then if the modulus of that you're going to measure is about in kilopascals, less than about 100 kilopascal, I will call it as a soft material. If it goes about 100 pascal, 100 kilopascal, I will call it as a hard material. So, like I said, for hard materials like bone and teeth and things like that, you have the typical harness tester, that nano harness tester that you can use, and you can use probably the well-established models to deconvolute the mechanical property. But when you go to soft materials, many of those things are not going to work. I'm going to go and point out a few things here. And then the reason being soft materials are being so soft, like when you bring a very sharp probe and try to make contact on them, you may not even sense the surface of the sample, and that is the problem basically if you use a sharp probe, because by the time you make a contact, like say that you know that you're sensing some force, you are already deep into the sample, and then in the assumption that you have the geometry and then there is a contact, size associated with the geometry, that will not hold good. So normally what people do is they will use a bigger probe, sometimes a very flat ended bigger probe, and then apply the force and measure the displacement. You can do that. You will have a still higher resolution in the vertical direction, about nanometer, but the lateral resolution goes up. Okay, so and also sometimes people do this experiment inside a liquid environment where you kind of, it minimizes the addition characteristic. So there are ways to do that. So what I mean to say is basically the methods and the techniques that you develop at measuring properties for metals and ceramics, it will be good for hard materials, but they are not good for soft materials. So you need to have a different way of thinking and different way of doing it. Right. So and also like when you do it on soft materials, for example, let's say you have a contact lens which is a hydrogel, and if you do a simple experiment with a typical instrument like 950, what you normally get is this kind of load displacement curve, which we are very familiar with, and we have a zero load, and then the load increases, and then load decreases. This is done at a displacement control. That's why you have vertical drop. But this is what normally you will expect to see as a load displacement curve. But in fact, if you allow to measure the full interaction of the probe with the sample surface, and if you do that, you typically get a curve like this, which is something very similar to what you normally get with an AFM, where when you bring the probe is coming closer to the sample surface. As the probe comes closer, depending upon the partition between the stiffness, whether the probe is having higher stiffness compared to the sample, or the sample has higher stiffness, local higher stiffness compared to the probe may jump into the sample, or the sample may jump into the probe. Okay, that's why you get to this jump to contact due to the addition between the probe and the sample. Then you further load, then you pass through the addition to a repulsive regime. Then you start to see this typical load displacement curve. When you unload, you have addition between the tip and the sample, and then you have this pull off. Pull off will give you a kind of a measure of the addition between the sample and the tip. Okay, so this is what normally you will get on a soft sample, soft biomaterials. Okay, and so you can't ignore the fact that you have all this interaction that is happening, and simply assume that it's kind of a typical load displacement curve and make a measurement. You will be in error. For example, in this particular case, you see that the jump to contact happens at this point, and that by the time when the when the probe goes to the zero load, you already penetrate into the sample about more than 2.8 microns. So if you assume that your contact just happened and there is no penetration of the probe into the sample, and that is the zero load and that is the zero displacement, you're making a big mistake in calculating the actual contact size. So and also, like if you look at here, this the attractive, the pull-off force is almost about 12 micronewton. If you look at the maximum force is about 12 micronewton. Normally we kind of ignore the addition forces are much smaller than the contact force that you apply. We ignore the addition part, but here you can't do that because the addition forces is almost comparable to the forces that you are applying. And you're talking about the length scale, about in this case is about from the point of contact to that maximum load is about 5 micron. So even for small forces like 12 micronewton, the displacement range is about 5 micron. So normally for soft biomaterials, this is what happens. Like you even for small, very small forces, the displacement range is much higher, and you need to have a very high force sensitivity to even sense the sample surface. So the whole instrumentation setup is completely different. You need to have high force sensitivity, very larger throw. So then we thought about that, and then we went back and we redesigned the transducer in a way that helps. I'll skip all these things, which is not important. So we call it as BioSoft bioindenter, and which is basically a transducer capable of applying very small forces with high force sensitivity, but also has a very large displacement range. And it has the same elements, like for example, the capacity sensor normally used in our typical high-strength turbo, and you still have that, but it is being used in a combination with the flexure actuator, an additional sensor, that you can go for a very large displacement range, about 150 micron, but at the same time try to keep the same force sensitivity that we have with the normal transducer that we use in our 950. Okay, so that.
Sorry to interrupt.
5 minutes? Okay, I'll try to accelerate.
No, like 2 minutes.
2 minutes, okay. So with that, we were able to put that on a biological microscope that without affecting the light path. Now, this is an inverted biological microscope. So when you use this, we can apply this transducer to apply force, but at the same time, you can also observe through this optical microscope as you bring probe in contact with the sample surface, how the contact is evolving. This is just a simple video where you can see this low displacement curve on the right, and then on the left, you can see this how the probe is making contact. This is done very fast, so you're not able to see how the contact is changing, but you have the ability to observe that through optical microscope, and the video is also synchronized with the force displacement curve that you measure. So all these things that I was talking about, making a pull-off force measurement, or making the stiffness and calculating the property through this low displacement, you can still do that along with this video image that we can correlate with. Okay, so that allows us to do experiments on materials like contact lens, very soft materials. And like I said, that if you do it in a liquid environment, you can avoid this adhesion problem. In those cases, this simple low displacement curves, you can fit with the Hertzian contact mechanics, and you can do that different rate of loading. There are two samples that I have shown. One is HEMA, which is acrylate based, and that silicon based contact lens. You can see the silicon based contact lens here has kind of a loading rate dependent, whereas in the HEMA, which is acrylate based contact lens, it doesn't have a loading rate different. What I mean to show here is basically you can do this even on materials like contact lens, which is basically a hydrogel, and reliably measure modulus of the material at any loading rate and make a good measurement. You can also do load relaxation type experiments where you apply a load and keep the load constant, measure the displacement as a function of time, or apply a load and keep the displacement constant and measure the load relaxation as a function of time. And then use this load relaxation curve to fit this part of the curve and measure relaxed elastic modulus. You can do that also. Okay. So another example that I would like to talk about is about the heart valve. This is very important. One of the problems with the heart valve is that the valve has to open and close, and sometimes if the opening doesn't happen, that leads to kind of a disease. And then the action of this heart valve is to pump the blood in one direction. So when the valve closes, it should close. It should not allow the blood to leak through. So this is kind of a mechanical function of the heart. And there are two types of valves that you can make. One is a mechanical valve, and the other is using a valve of animal tissue, using tissue engineering. If you use a mechanical valve, it will work for a very long time, but what happens is the blood will float onto the valve, and you need to always take this blood thinning medicine throughout your life. But if you use the tissue engineered valve, it has a very low life cycle, maybe about 10 to 15 years. So if you wanted to do this tissue engineering properly, you need to understand how the material property is supposed to be. So in this particular case, if you do an indentation experiment at the periphery where that opening happens, it should be more elastic. At the point where it is anchored, it should be more rigid, and the middle in between. So if you go and measure these properties at 3 different locations, you can see that the periphery where it is supposed to be rigid, it is very stiff. At the periphery where it's supposed to be elastic to have conforming closure of the wall, you can see it is highly elastic. So using this kind of technique that we have, this indentation technique, we can go and probe and understand, and then we can design materials accordingly. So I'm going to, there's one more example. Do you have a minute, Pratyang?
We already
okay,
exceeded,
then okay, so I will just go and this is one case where we can compress a cell but I think I just, for the sake of saving time, I'll just go ahead and stop over here. So with this BioSoft instrumentation, you can do high force sensitivity with also very large displacement range. That we can use it on a biological microscope, which is very useful for understanding properties of soft biomaterials. With that, I will stop and take any questions.
So yeah, so Doctor, due to time constraint, what we are thinking, because the next talk is from Yogesh again on the AFM perspective. So we can take the questions together, and because obviously after the next talk, there will be maybe a question on which instrument to go for when you're testing on biomaterials and where they are more useful. So after Yogesh talk, we can take the questions together.
Very good. Yeah, that's fine. Good.
Okay, so I'll just share this.
And that's also a good thing. So all the tough questions Asif can take.
Yeah, no problem.
Absolutely.
Yeah, you know that I am in Minneapolis. I probably go to sleep.
Yeah, it's midnight there. Already past midnight.
1:45 a.m. here.
Yeah. So, okay. So with that, I will just go to our next speaker of the day. I mean, the last talk of the day as well. Dr. Yogesh Jairam. He's currently working as a key accounts manager at Bruker India Scientific Private Limited. He has graduated with a PhD in solid state physics and magnetism at Catholic University in Leuven. He has mostly worked as an application and as an engineer and scientist with Bruker, but with experience and exposure that he got, now he's working as a key accounts manager, managing all the sales account for AFM as well as Nano IR, SOM, basically stylus profilers, and TMT product line. So without further delay, I would call Yogesh to present his talk today.
Thanks, Pratyang, for the introduction. So let me share my screen now.
Sure.
All right. Can you see?
We are seeing more like a grid view. Yeah, now we got the slideshow.
All right. Yeah, better?
No, yeah, now it's okay. Now it's okay.
Okay, that's good. Then, all right. Hey, good morning, everyone. So many of you, maybe it's time for lunch. So I'll try to keep mine short and sweet so that you all have a good lunch break. Today we saw a lot of different talks on doing mechanical properties for biological materials, right? Like bones and cells and tissues and different properties. So I'm going to focus on the same theme, but by using mainly atomic force microscopy and doing different mechanical testing using that instead of a conventional nano indentation. And we will talk about the advantages and disadvantages. Of course, I think Professor Balani also talked about it briefly earlier, right? So if you look at the AFM, so you have the cantilever and you have the laser that's getting reflected from top of the cantilever. Let's see if I can get, yeah. So the laser comes and then it goes to this 4-point detector. So the cantilever moves up and down. That means this point, the laser point here on the detector will move. And based on that, you have some clever electronics. So then you understand what's the kind of topography there on your sample. And because here you have the probe is contacting with the sample, you can also get mechanical information, right? So the most important thing in AFM, right, is the force distance curve. So it's a little bit different from the nano indentation point of view. So maybe I'll walk through here. So the key differences are here you have the cantilever and then end of the probe is there. So these are typically like silicon nitride kind of material. Of course, you can have different materials there. So the tip is very small, right? We are talking few nanometers. So let's say if you're very far away from the sample, so this is the sample, you're very far away, the force on your tip is 0, right? There's no force there. There's no force on the sample. Nothing is there. Then as you bring the tip closer towards the sample, so that will be the red line, you follow the red line, you come closer to the sample, then you have basically what we call the snap-in contact. So this is the attractive force exerted by the sample on the tip, or so then the tip will actually bend a little bit forward and then will touch the sample. And then you keep pushing the cantilever further down. You push this down. Then basically you have like a cantilever bend, and then you increase the force on your cantilever also on the sample. So now when you pull the cantilever out, so you start retracting it out. So you retract, but it doesn't come out here because now you have the adhesion force between the sample and the tip that's holding the tip longer. So now you have this kind of bending of this cantilever in reverse. So then it takes a little bit more pulling to get it out of the contact, right? So this is basically the adhesion force between the tip and the sample. Then again, once you are very far away, there is again zero force here. So you go back to this basic step. So this is the force distance curve, and very key when you are talking about mechanical properties, right? So it's the actual physical contact between the probe and the sample lets you probe the mechanics of your material. Then there is, of course, the scale. That's one of the major things for the AFM is it can have very, very high lateral resolution. So here we say from atoms, so you can see nice atomic resolution imaging. You can see the lattice and maybe some defects here, etc. Right? So then you go a little larger scale, maybe like single molecule, like DNA. If you look closely, you can see the major groove, the minor groove, right? And these are single protein. This is a BR trimer that you see. Then you go up the scale in several microns, you have the cell, and you can see the finer details of the cell structure, like the microvilli projections there. And then you can go further higher, right? And then, but that's typically outside the range of AFM. So yeah, so from atoms to cell, this is really the range in terms of what you can actually measure with AFM. Yeah, so that's basically the cartoon for that. And we can also now translate that in terms of what would be the modulus, Young's modulus, and what would be the range that we can probe with AFM, right? So yeah, for sure, the softer materials like cells, molecules, etc. And then if you go harder materials, maybe like collagen, can be several 10 gigapascals or so. These are also possible to probe with the AFM. And of course, when you go for the harder material, there will be a lot more error compared to the nano indenter, but you can still get a good understanding of the at least the relative contrast on your samples. And yeah, so this is all we can do. So AFM, when we work with biology, when we develop these instruments, we put a lot of emphasis on it working well together with microscope. So what you see here is a microscope image, and you can see the AFM cantilever here. So this is the live cell. This is under liquid or the buffer conditions. And then you are basically, you can draw a box where you want to image. So here we are imaging almost at the cell boundary here. Then you can just draw a box there, and then you can start imaging there. So now you see the microscope and then the AFM image. It's very important to have this kind of facility because, you know, as a biologist, many biologists working on this kind of material are used to doing microscopy, and sometimes it's also needed, right? So for example, if you have a cell, it's sometimes very hard to see on a simple bright field microscope. So you might need a phase contrast or a DIC contrast to find out the cells of interest before you go imaging it. So you need to have the AFM and the microscopy work together. And we put a lot of efforts in developing these kind of easy solutions. So you can basically draw boxes on your microscope camera image, and you can start collecting it. And I'm not sure whether you noticed, you also collect different channels. So here is only topography that's overlaid on top of your microscope image, but you also have the mechanical properties that's collected simultaneously, like modulus, dissipation, adhesion, etc. Right? So you can do that kind of imaging. So yeah, again, just a little bit of a static image here. So you can see this MDCK cells. And this is with the confocal, so you can see this kind of confocal or fluorescence image, and then you can draw a box, and this is the region of my interest. Then the AFM will go there and do the scan, and you can have some correlation with your fluorescence or your confocal image so that you understand your samples a little bit better, right? And identify the regions where you want the AFM to probe as well. Yeah, and this actually expands the area of microscopy. In microscopy, the traditional ones like confocal or fluorescence, you're only seeing, but you don't have any mechanical impulse. Now with the AFM, yeah, so you can use AFM as a static way of just for imaging, but also you can use AFM to give some stimulus, like mechanical stimulus, like pushing or pulling a cell and see how it reacts, how the fluorescence correlation work, and you know, Fred, those kind of analysis can also be done. And this is fully integrated with that. So let's get that out of the way. Then let's now talk about why we use AFM for nanomechanics, right? When we say you have nanoindenters, which are very good and have good resolution. So why we need AFM is, number one, if you want very high lateral resolution. So like I said, the AFM probes are very small, so you can get much higher lateral force, lateral resolution. And you also get very good force control, right? So you can now even control the force to 1 piconewton level. So this is possible, and it might be required, especially for some delicate materials. So here is a good example. So this is there are a bunch of publications on this. So this is a DNA that's imaged. So you can have a cartoon also overlaid to give you idea where the minor group, the major groups are there. And what happens is that when you image it with very low force, you can clearly see these nice structures there. But when you even go for like the 200 piconewtons, is still very low force, right? Yeah, then you already don't see this nice finer structures there. So it's very important to have this kind of resolutions there. Again, for mechanical properties, so here is a different example. So you can see there are certain features that you will see in mechanical contrast, but not in the topography. So it's also important to get very high lateral resolution for mechanical properties there. The drawbacks, yeah, of course, there is a modeling error. So there is a lot of different models when it comes to AFM and doing this, how to exactly extract your mechanical properties. So there can be 10 to 20% error depending upon what kind of samples we are using, right? But this can be minimized. But if you're comparing sample A to sample B to sample C, then it can also be even further minimized because you are doing a relative comparison, right? So this is, of course, something that we need to know about. When we go to hard materials like this, like collagens or bones or other things, maybe it's difficult to find a very stiff cantilever, right? Nanoindenters, you know, the stiffness of the system is very good. So this sometimes can be a challenging as well. So these are some drawbacks there, but yeah, so this is something we have to live with if you want the lateral resolution, right? When it comes to like, the AFM and mechanical properties, there are many different ways to do it. So, you know, when you do the nanoindenter or like the DMA, you're doing very low frequencies or pretty much like a quasi-static cases. But in AFM, you can have different techniques that also says that the AFM mechanical property mapping is still evolving, right? There are a lot of different ideas. There's a different hypothesis with plus and minuses. So the traditional force spectra, the one I show you a cartoon of before is somewhere here, the force volume. So very low frequencies again. But maybe go up to a few hundred hertz or so. Then you have the peak force tapping. This is a Bruker innovation, which lets you go maybe a few kilohertz. Then you can also do in the contact resonance mode, which of course is dependent on the sample. So what's the resonance of that? But it's very typically very high frequencies. Yeah. So that is also there. So it's very important before we start the mechanical properties, what's the right mode we want to choose? Because, you know, especially viscoelastic materials, the frequency of your modulation will determine what kind of modulus you get, right? So because they are viscoelastic. And this is very important that we nail this down before we start. And maybe in some cases you don't have a choice, but this is something important to know, at least where we are and what kind of results we can expect and how we can compare them. Then, of course, another major factor in doing is how do we know what's the contact area, right? How do I get the correct number? If you see the more typical AFM cantilevers are all like a cone shape, right? That's typical. And that means it is not going to fit very well with the Hertzian contact models. So it's not a sphere, right? So you're going to have either like a Snedden or something, and you can use that to understand different contact geometries. Of course, the frequency you use will also choose the different kinds of modeling you use. Then you also have to calibrate it every time because every probe is unique in the terms of the spring constant of the probe is going to be different. The tip is going to be slightly different from one to other, right? So those kind of things. But if you look at the overall things, I will not go into too much details about it. But here, the major error when it comes to the evaluation of your modulus basically comes from the radius of your tip estimation and the spring constant area estimation or calculation, right? So these two will play the major part there. So this is very important to get this corrected. So now to account for this, we have introduced several new lines of probes where you can have like a very nice defined radius of curvature, so as close to Hertzian as possible. And these are also pre-calibrated, so you exactly know what's that particular probe's spring constant, resonance frequency, everything, right? So very well controlled. So this helps. So here is an example, yeah. So if you choose the wrong probe, right? So you get the radius corrected and the spring constant. If you get the correct number, then it's good. If you get them wrong, the errors can be significant. So if you say, for example, I'm measuring this is the modulus and this is the predicted error. So if you choose the wrong probe, your error can be as high as 50%, right? So it's very important we choose this correctly. So based on our estimation of where the modulus we are going to do is going to come, right? So that's also very important when we do that. Okay, so this is the basic kind of idea. So just give you some setup, how the setup typically looks like. Not sure how many of you are familiar. So you have like a microscope where you can see through the binocular or the camera, the different contrast, whatever the feature your microscope has, DIC, phase or whatever. And the blue region is the AFM and your sample sits on this blue stage. So the AFM will be here and then you can microscope. You can have both the data collected in a computer, right? So this is basically roughly the setup. And this is very adaptable. So you can basically fit this kind of AFM in any different microscope. So here we use the Zeiss, but it can be as easily accommodated on a Leica, Olympus, Nikon, you name it, right? And very flexible. So it uses basically the IOM condenser, objective, etc. So any optical mode, right, can be easily done. Even if you're doing like FCS or FCCS kind of microscopy is still compatible. Okay, some simple things here. So here, some example. So what you see here is a cytoskeleton. So you're imaging a cell which is living. So you have the cytoskeleton reorganizes as the cell moves. And you take an image at different intervals. So after 5 seconds, after 11 seconds, etc. And you can see there are some movements in the cytoskeletons, and we can model them. And this will also help you understand what the force displacement fields and how they move inside a cell, right? So help understand fundamental level how the cells behave and what kind of forces are there and how they move, right? Okay. Can you hear me?
Yeah, we can hear you, Yogesh.
Oh, okay. For some reason, my computer got stuck, so.
Okay.
I don't know what you can. Oh, sorry. Oh, sorry. Yeah. All right. Eventually, it comes down now. Okay. So you can see my slide, right?
Yes, please go ahead.
I'm on 16, yeah?
Yeah, cell tissue substrate mechanics, right?
That's perfect. That's the slide I mean. Good. Okay, so like we talked a lot about the force indentation curve. You can also do something very close to like nano DMA on your cells. You come contact and then you modulate it once in contact, and then you can look at the dynamic performance of your material, right? So here are some really cool examples. So this is a red blood cell, and you can see these are the spikes on top of your red blood cells, right? So this is actually due to the malaria infection. So your healthy red blood cell is nice and smooth. Once it's infected, you get this kind of spiky structure on top of your cell, right? So this is how the cell looks like. So now if you want to have, let's say, while you're developing a drug or something, and you want to make sure that it sticks in particular to a infected cell, not the healthy one. So you need to identify which is the uniqueness of the infected cell, which is the healthy one, and you need to go and attach yourself there, right? So this, so what we have done, the researchers have done, not me personally. So they have done in this case is they have taken a cantilever and they have attached the CD36 to the cantilever. They use another in-between molecule called PEG linker to attach it. So they attach it there, and there's a lot of procedure to do that. And once they have done that, they go and image with this modified cantilever, right? So when you image with the modified cantilever, you can see very nice things. So this is the topography, and the brighter regions means higher, right? So you can see this is a little higher. So these are the spikes we saw on the cell, right? And when the CD36 is attached, you also see that there is an adhesion is very high on top of this spikes or knobs, or how you want to call it there, right? So that basically means that this molecule likes to attach here more than the other regions, right? So when you use this kind of molecule, that targets basically the infected red blood cells over a healthy cell, right? So that's that side here. So another region where the AFM really excels is probing single molecules. So you have a single molecule. I want to image them. You already saw some images before of DNA and trimer, BR trimers. And if you want to also understand the mechanical features of your molecule, right? Either like binding, unbinding, or what's the addition, right? So those kind of things, you can use the AFM cantilever to do it. So here are some examples. So here's like basically ripping the DNA, and you see what's the energies there, what it takes to damage. And you also have like a long protein, and you can attach it to the cantilever and start pulling it. You can understand this. So different kinds of this. So then also you have different molecules. So you have one molecule in the cantilever, the other on the sample. So what kind of interactions they have, right? Or simple protein unfolding, right? So that's kind of different studies there. So here's a simple example, a very popular one. So this is a tintin molecule. So this is a very large molecule. So you can, when you start pulling it, so you have this a big drop, right? So this is basically when there is a subunit that unfolds. So you have a subunit that suddenly unfolds, you will have a big drop. So you have the force, and you're pulling it, and then you have the several levels of drops, right? So this is basically how the force distance goes. So you are pulling it from 0, and then maybe it completely unfolds at 220 or 230 nanometers. So very, very long molecules. And then you have about a subunit that's unfolding every 25 nanometers or so, right? And you have the worm-like chain model. It's one of the very popular models that people use to study these kind of unfolding. You can use that to fit it and look at your protein. Many times you might also be developing the models or different versions of this. Then you can also use that to study here. So this is a very particular example. Another slightly larger molecule was published by Kedrov and his colleagues. So here you have NHAA molecule, right? And you are basically looking again at unfolding event. So this is like a representation of the molecule, and then you come and you attach it here, and then you start pulling it. So from the modeling, we know roughly what these are and what kind of where they will unfold, etc. And we can correlate it, right? So this is your recorded data, and from your theoretical predictions, you can correlate it and then improve how we model these kind of molecules, right? So that's how we use this kind of studies to help advance our understanding of this at a fundamental levels. Now I'm basically just going to slightly larger scales. So here, it's a very cool experiment they did here from Nature Neuroscience. So they take a xenophus. I think this is basically like a frog, very like a embryo stage frog. They keep it alive, but they open the head. Yeah. So they peel open the head, so you can see the brain that's developing. So this is still alive. So this is kept in alive conditions, and they come with the AFM. And they look at how the axons are growing, or RGC axons, and how the mechanical substrate or the background in which they grow, and so this is something that they did here. So maybe a little bit background here for people not very familiar with this area. So it's been known, like for example, like if you have like a stem cell that's growing, depending upon the substrate and the modulus of your substrate, your stem cells will go into different ones. So if you have a very hard substrate, they generally go into like a bone cell. And if you have very, very soft substrates, it will go into like a neuronal cell, right? The stem cells will, and so you can basically say there's just not chemical signals that's there in the biological world, but there's also mechanical stimulus that basically direct how the growth of the organisms or how they behave. They also take mechanical cues from the surroundings. And this is what they are basically trying to probe in these kind of studies. But here they are looking at a living organism, quite a large one, right? And then looking at it, how these things are. Yeah, so this is basically whereas as the brain is growing, there basically will appear stiffness gradients inside, and you can there will be, yeah, this is just a modulus map you see here. There will be stiffness gradients, and they basically notice that the neurons grow towards the soft tissue. So the gradients are there where there is things are softer. The neurons will generally go towards that. So that's basically the finding that they did here, and it's very clear, right? And it's in agreement with the other studies where they look at cells individually outside of the living organism, but this is the one. Earlier, I think we also hear about in the talk from Professor Balani about the additions and to different surfaces, right? The cell additions are very complex things. So this is the simplified version of it, right? So the simplified version of it, so you have different things. So you have integrin or caterin or vinculin, etc., etc. So you have the cell friend there, then you have this kind of molecules, you have addition there, right? You have cytoskeletons, focal complexes, etc., right? But what is really happening in a cell adhesion is very complex. So you can see here, I think this is quite old paper. So 2001, probably this chart is even more complex now. There are so many things involved, right? When we look at how the adhesion of a cell is driven, right? So how the cell adheres, how the cell moves. So this is the cell contact. So it's very, very complex studies. And there are a lot of people looking at it and trying to expand our understanding of that area. So, when it comes to the AFM, so you can basically look at this way. So what we see here is a cell. So this is like, I think, one of the xenophus cells only. and you can come with a spherical indenter. So it could be like a glass bead or polymer bead. And this is the side view, right? So you're here looking from the side. So this is the bead, this is the cell, and this is the probe that's holding the bead. So you're looking from the side, and this is you're looking from the top. So, okay. So now let me come out of the pointer and play you a video. So this is basically called blebbing. So what is happening is there is some internal pressures in the cell that create these kind of blebbing membranes, and they do this kind of behavior. And this will also affect how the adhesion and other property works, and also the hardness of the cell interaction. So cell, you see, is a living thing, right? It's not like a static case. So I indent here is always going to indent at the harder region or the softer region of cell. No, because the cell is constantly moving, is doing something. So you can end up in the same spot, but you might hit the bleb, or you might hit the other regions of the cell, so you can get a different property. So again, you see here, this is from the top and this is from the side here. So you can see this kind of behavior, right? So here, and this is basically combining these two. You are coming from top, and then you are going to do the indentations, and you are going to do many indentations, and you can see whether you land on the bleb or on the other regions, you are going to get a different modulus, and some very different ones at that. So you can see here, the red dots are when you land on the bleb, and the blue dots are when you land on a region that's not the bleb. So you can see here, the non-blebbing regions are typically much stiffer compared to the blebbing regions, right? So you can do this many times, and this basically helps. So if I had only this graph, right, when I look at the top view optics and I have only this graph, yeah, I will be just confused, right? What's happening? So now once you can correlate it well with your side view optics, okay, now you understand a little bit more, right? Okay, what's happening when I get the higher modulus or when I get the lower one, right? And you can still see there's a lot of standard deviations within that as well. So that is the idea about giving you different information from different directions, right? From microscopy, conventional one, side view microscopy, mechanical information. You basically help understand what's happening in your material further and further. Yeah. So now basically we take that and we make it more complex. So now if you look at a cell-cell interaction, same kind of cell, one is on the top, one is on the bottom. And what we are trying to look here, so you can see these are basically here two cases. When the cell-cell interaction happens, when the adhesion is mainly from a non-bleb region, or if it's attached to the bleb, you can see there will be a very different adhesion force. So it's basically here they say it's a detachment force. So you can see in this case, it detaches much earlier. So you will see already gone. So that's because it hit the bleb, right? Then the bleb basically promotes cell-cell detachment. So when it's stuck to the non-blebbing region, your force is much higher. So 15 nanonewtons to basically 4 nanonewtons, right? It's a huge difference there. Of course, there's a standard deviation again. So, but that you can see here, this is something very important. So you need to not only do a force distance curve or look at the graphs, but you also need to see what's happening so that you can get a little bit better picture, understanding of your materials. So that's roughly it. Maybe I overshoot a little bit, but yeah, I think I finished it in time. So if you have any questions, we can take that.
So yeah, Yogesh, so thanks for the talk. But we have a question. Since most soft biomaterials are viscoelastic in nature, how can the AFM load displacement curve be used to yield stress strain curve and viscoelastic properties?
So yeah, so like we said, in AFM, used for many different purposes, right? Sometimes you are just understanding, like the example I showed in Xenophor's brain. You're not really interested in exact number. What's the modulus? What's this? What's that? We just need to see how the gradient is, if the region is softer or not. But sometimes, of course, you are interested in the exact properties. Then, of course, you have to choose the right technique and other things. We can play with that. And there are different models also that help you understand your viscoelastic properties. Like we said, there is also something we can do like NanoDMA, which Dr. Asif is a much better expert than me. So you can talk about that if you want. You can do that and to get a little bit more precise data. Of course, your contact is now through AFM probe, and there are some things that you need to account for in terms of the softness of the probe, etc. But you can use the AFM to do that. Yeah.
Right. Okay. So just a generic question here. I mean, after we are, finish all the talks today, and now we have two tools that we are seeing, Nanoindenter and AFM. So which one, for a user point of view, one can give you a large displacement, one gives you better lateral resolution. So how one has to choose, which tool? We are giving a complete solution for biomaterial characterization, but it's an ultimate question at the end of the, the event. So that's the message also we wanted to convey, clarify on this. So maybe you and Dr. Asif combinedly or can answer on those aspects.
I think it's not like well, this is better than that. I think you are going to get different information from different techniques. The more information we have from different dimensions or different directions, it's going to help us enhance our knowledge of what we are studying, right? And same with any microscopy. That's why you just don't like, okay, I have FEM, I don't need optical microscopy. No, that doesn't work like that, right? So you need different techniques at different point in time, depending upon what we are studying, what we want to get at. And maybe sometimes we need more than one technique to understand what's happening in your material.
Yep.
So to add to that, it also boils down to the length scale of interaction that you're interested in studying. When you go typically nanometer to higher micro scale, you need to use a different type of instrument compared to when you wanted to study around DNA level or submolecular level, you may have to use AFM. So it depends upon the length scale also. It dictates what instruments that you probably have to use.
Yeah.
Right. Okay, I think I'm not seeing any further questions there. So I think, yeah, it's pretty good. So thanks, Yogesh, for the talk. And with that, so with that, I would like to first thank all our speakers of the day today, Professor Naveen Kumar from IIT Roper, our keynote speaker from IIT Kanpur, Professor Kantish Balani, and obviously Dr. Asif for staying with us also for the entire event. It's almost like maybe 2:30 there now, 2:30 a.m. So thanks for staying with us for the entire event, and Yogesh for a wonderful talk on the AFM perspective. So, but nonetheless, to have a successful event, we need to have a good participation from the attendees. So big thanks to all attendees for joining us today. It's a field which you are trying to also reach more people, because it is definitely serves a purpose. It helps you to know more on the technical side or characterize your biomaterials. For any further inquiries, you can always reach to me at pratyak@industron.nano.com. We are based in Trivandrum actually, and we also channel partner of Bruker. So any query you have with respect to material testing, application, or even product information, please feel free to reach us. Also, we have also
a nice lab in Trivandrum. So
that's a quick glimpse of a lab, which is equally with the nanoindentation systems. Also, a lot of queries, people have asked us about the recording. So yes, there will be a recording of the session. We will be posting on our event page. We will send you a link to all our registered participants, even participants who have missed to attend today. So with that, we conclude our session. And again, once again, thanks to everybody. Stay safe and hope to meet soon. Okay. So,
all right. Thank you, everyone.
Thanks,
Thank you.
everyone.
Thank you all. Bye-bye.
Bye. Take care.